Very good morning to one and all. I welcome Dr. Saurya Dattagupta. Indeed, I am very happy to introduce Saurya Dattagupta sir, as he is one of my teachers, my professor's son. Actually, Professor Dattagupta is one of the famous physicists in Central University of Hyderabad, especially in laser physics. Till today, I have his notes, running notes, and all other things. When I remember, so I'm very happy to introduce Saurya Dattagupta sir to this gathering here. I request Shainita to introduce to the participants Dr. Saurya Dattagupta.
Good morning, one and all. Now I introduce our today's speaker. Dr. Shaurya Gupta is currently working as an assistant professor in Material Science and Metallurgical Engineering, IIT Hyderabad. He is also a visiting assistant professor at Cornell University between May 2018 to July 2018. He has obtained his postdoc from Applied and Engineering Physics, Cornell University between August 2016 to July 2017. Postdoc from Department of Physics, UT Austin between May 2015 to July 2016. PhD from Nanophotonics and Meteorology Lab at Swiss Federal Institute of Technology, Lausanne, in Switzerland between July 2010 to April 2015. He was awarded with Ramanujan Fellowship in 2000, Distinguished Referee Award by EPJAP in 2018. He has published 20 papers in international journals and attended 18 invited talks and conference proceedings. His current research interests are surface plasmons supported by alloy thin films, template-driven assembly of anisotropic nanoparticles, graphene integrated metasurface devices, mode analysis, fabrication and characterization of plasmonic nanostructures. He has funded research projects from the Nano Mission, SERB Early Career Research Grant, SERB Ramanujan Fellowship. Now I request Dr. Shaurya Gupta sir to start his lecture.
Thank you very much for this. So here and to give this talk in this gathering. So as you might know that the topic of my talk today is plasmonic metasurfaces for biosensing and their potential for disease diagnosis. So as was mentioned, I am a faculty at the IIT Hyderabad in the Material Science Department. So this is a typical evening sunset picture of IIT. So it's a very pretty site. So if possible, please do visit us. So the outline of my talk will be as follows. So first I will talk about what are these plasmonic metasurfaces that I have put in the title. Then I will talk about how we can use these plasmonic structures for either doing surface enhanced Raman scattering or for distinguishing between cancerous and normal cells, and then their potential diagnostic applications. Right. So let us begin with why do we really want to go to the nanoscale, and that is the whole point of this whole workshop and the short-term course. Right. So if you take bulk gold, it has a nice yellowish color, which we all are very familiar with. However, if you actually reduce the size of gold, what you see is you can get very vibrant colors ranging from red to blue, right? And these colors are actually strongly dependent on the size. So if you change from around 2 nanometers, that is on the left-hand side, to a color which is bluish because of a size of around 150 nanometers, you can get all these different variations of colors, right? So this is what makes gold so interesting at the nanoscale. This is just one example of how the nanoscale properties are so different from the bulk properties. So now let's come to the question of what is plasmonics? Now, plasmonics is basically an area that deals with the interaction of light with metallic nanostructures, right? So let's say you take this gold cuboid. Now, in this gold cuboid, if you send light, and now light we know has a certain electric field associated with it. So if that electric field is along this length of the cuboid, and if you put a detector which is very far away, right? You just put a light detector far away. What we will see is that at some wavelengths or frequencies of the incident light, the intensity will be highest at the detector. So if we plot it, it is shown here. So the horizontal axis is the wavelength of light that we are sending in, and the y-axis is the scattering efficiency or the intensity at the detector. And what we see is at this wavelength of around 700 nanometers, you have a peak, right? So the scattering is, let's 20 times higher as compared to other wavelengths, the non-resonant wavelengths. Now, if you actually notice, don't put the detector very far away, but you actually come very close to the structure, to something that we call as the near field, and you look at this particular wavelength, you will see a plot like this. So the color here, the from blue to red, actually represents the electric field amplitude, right? So blue basically means a low electric field amplitude. Red actually means a very high electric field amplitude. So now, if you notice around this structure, around this cuboid, what we have is at the corners, we have very high field enhancement, right? It goes to almost by a factor of 14 as compared to the one which is farther away, right? And this is the field, the near field is what we exploit for various applications. Now you can talk about applications in diagnostics and therapy, or you can talk about applications for optoelectronic devices. Both of these are possible with these plasmonic structures. So now the simplest thing that you can think of is, can we make a simple sensor? Now, let us say I give you an example that you take this cuboid, you place it in water. So the refractive index of water, which is a basic property of any medium, is around 1.33. And if I change that water medium to something, let's say water plus glucose, so the refractive index will increase and go to 1.4. Now, when this happens, if you look at the resonance with the same geometry, the same structure, everything, the resonance, you will see a shift. So the blue one corresponds to the case when it was water, and the red one corresponds to the case when the background was water plus glucose. So you see this shift. So by looking at this shift, we are able to say what is the change in refractive index. And thereby we can actually go back and say, if it is a water glucose mixture, how much of glucose is there in water? So this is the simplest sensor that you can make using plasmonic structures. Now the question is, can you do something more than just these simple sensors? The answer is of course yes. And the reason is very simple. If you, let's say, talk about interaction of molecules with plasmonic structures, the interaction between molecules and light is proportional to the electric field squared for absorption and electric field to the power 4 for Raman scattering. Similarly, fluorescence and IR absorption also get affected based on the electric field. So now if I just look at this simple variations, let's say this is the electric field modulus. So if I put my molecule far away from the structure, it will not absorb a lot of light because the electric field is low. However, if I take that same molecule and put it near these corners where we have the high electric field, the absorption will be enhanced by the electric field modulus squared as shown here, right? So if I put the molecule here at this corner, my absorption of that molecule, that same molecule, will be enhanced by a factor of 140, right? So what this allows us to do is we are able to enhance the signal from the same volume of or concentration of molecules, right? Now, another thing that you will notice is if I have molecules here, only then will I get a strong signal. If I put the same molecule here, I'll get a very low signal. So what I'm also doing is I'm reducing the volume of the molecules or the concentration of molecules over which I'm getting the signal from, right? So these are two common ways that you can use plasmonics for doing sensing, which we'll come to later. Now, as I said, it's a resonant structure, right? So how do we tune the resonance? Now, one way of tuning the resonance is you change the material that you have, right? Standard materials that we work with are gold, silver, copper, all the precious metals that we can think of. Now, people have started working on new materials like titanium nitride, aluminum nitride, or alloys based on copper, silver, gold, right? This is one possibility. Another possibility is why just work with cuboids. You can actually change the shape of the particles, which are very nice way of changing the resonance. For example, these are the three shapes that I'm taking here: a cuboid, ellipsoid, and a cylinder. Now, this is the scattering that comes from these three different structures. This is a wavelength and this is the scattering intensity. We see that they showed three different resonance peak positions, right? So you can control it this manner. Now, the third way is why only single particles. You can start assembling these particles into different arrays. This is like, for example, 7-particle array. This is basically a dimer where you have two cuboids, right? So these are simple ways that allow you to actually tune the resonance of the plasmonic structure, right? So, and this is basically a major part of plasmonics, that you want to tune the resonance that it's optimized for your particular application. So now, let us take this case of a plasmonic diamond. So we have two gold cuboids. So both of them are made up of gold. This size is around 60 to 80 nanometers, and the gap we can vary. Now, if I have a gap of 20 nanometers, I see that the field, which is shown by the color, so red is a higher field as compared to blue. As I reduce the gap, the field increases, right? So if I have to use it for surface enhanced Raman scattering, where the signal is proportional to e to the power 4, I would prefer a geometry which has smaller gaps as compared to larger ones, right? So a practical example of this can be seen in this structure here. So what we have here is, we have a kind of a gold grating. This is the SCM image of the fabricated structures, and this is a schematic, right? Now this structure also has a gold layer at the bottom and a certain dielectric layer in between. Now we are sending light from the top and looking at the reflected light. Now if I look at the reflection as a function of different values of this spacer of SiO2, that is shown here. So the left column is basically the theoretical calculations, and the right column are basically the experiments. So the horizontal axis is the wavelength and the vertical axis is reflectivity. Now as I change this spacer from 100 nanometers to 700 nanometers, I see that the reflection actually changes significantly, right? Now these two dashed lines that I have correspond to the laser excitation wavelength and the laser emission wavelength for the Raman process that I'm looking at, right? So if I take this particular value of D, I see that the structure is non-resonant, that there's no resonance at this particular wavelength. However, for this particular case where D is 150, I have a resonance at both the values or close to both the values. Now, if I compare the fields, comparing this particular case, I see that the field is very low. So it's all blue colored. However, for this case where it's resonant, I see that you have a lot of red regions, which we call the hotspot regions. Okay. So, and correspondingly, if we measure the Raman from this molecule, mercaptobenzoic acid, we see that, look, as for certain values of D, like for example, here 300 nanometers and 700 nanometers, the surge intensity or the Raman intensity is very high. However, for some other values, like for 100 and for 500, the values are very low. Okay. So what I'm trying to emphasize here is that by choosing the structure properly, we're either able to enhance the Raman or completely suppress the Raman signal that we are getting. Right? So now let us come to what are plasmonic metasurfaces. So plasmonic metasurfaces are 2D arrangements of these structures that we talked about. Right? For example, here we have an array of multiple C-shaped structures with a wire running through. Right? So you have a sheet of these structures which you can make using different processes. These structures were made using a process called as electron beam lithography and lift-off process. Now you might ask, where can we use these nanostructures for different applications? You can use them for sensing, you can use them for optoelectronic devices. And this are some different designs that people have made. So you can make cylinders, array of cylinders, you can make array of rods, you can make V grooves, V shapes for different applications, right? Now, this is another example where they have used this kind of structures for doing biosensing. So what they're doing in this particular work is that they're looking at the orientation and the thickness of a single layer of molecules, right? So just remember, a molecule that is used in this particular study is around 2 to 5 nanometers. So it's very, very small. So here what they're doing is they're precisely measuring the exact thickness and the refractive index of that single molecular layer that they are putting on the structure, right? So let us come to the first part of the talk today, which deals with a single nanoparticle analysis using SIRS, right? So question is, why are we interested in nanoparticles and how does it come to diseases, right? So now the way it works is, let's say if you use nanoparticles in a day-to-day life, for example, in sunscreens or in for cancer therapy, which people actually use these gold particles, the nanoparticles have to be put in blood or some kind of body fluid, right? Now, as soon as you put these nanoparticles in a bodily fluid or some biological fluid, what happens is the proteins in that fluid actually come and stick onto your particle, right? So some information from that fluid is actually encoded in this protein corona, right? So let's say I take these particles, let's say gold particles, and I put them with cells. In this case, we have used human cells. and I leave them for, let's say, 96 hours, these particles will be coated with a layer of proteins. Now, this layer is called the protein corona, right? And now this protein corona determines how these particles will actually interact with our body, right? So it is very important to understand this layer of proteins, right? So the standard way of doing it is you typically take these particles, try to put it through a mass spectroscopy instrument, and then you get a list of proteins that you can analyze. However, you have to notice that this is very static, and mass spectroscopy is typically a destructive technique, and you don't have single particle kind of information, right? So because every particle will be different, so you want to study at a single particle level. So this is typically kind of the data that you get. So you'll have thousands of proteins that you get from mass spec, and then only choose the proteins of your own interest. So here, if you notice, this is basically lots of diagrams, but it's very simple to understand. So horizontal axis are different kinds of nanoparticles that we have used. So no nanoparticles, two different kinds of nanoparticles, and the vertical axis is different kinds of cells from the human body which are cancerous, right? And what we see is not all the colors are uniform. So some are brighter than the others. So if I highlight it, so these are the ones which have the highest contrast, which indicates highest particle uptake by these cells and this combination. So what we see is this nanoparticle uptake is not uniform, right? So what that is telling us is the type of nanoparticle matters significantly. And that is precisely why we want to understand how this protein corona affects, right? But that is very complicated. So as a as an easier system to study, what we did was we synthesized particles with a known protein corona, right? In this case, we are using this molecule called a cytochrome C. Right? This is a molecule that is commonly found in the mitochondria of all organisms, including humans. Now, this protein has a size of around 3.4 nanometers, right? And this is positively charged at neutral pH. So what we do is we do a simple nanoparticle synthesis where we reduce the gold salt using HEPAS, which is a buffer, and add cytochrome C at the same time. So we get gold particles coated with cytochrome C. Now, this is a typical size distribution that we get. So this is the size, horizontal axis. Vertical axis is the histogram. And if you notice, we get nanoparticles of around 12 nanometer in size. And as I had mentioned before, small size gold particles have a typical red color, which is shown in this append, which is the second one, right? So now the idea is, can we analyze these particles to show that we have the cytochrome C molecules on these particles, right? So typically, if you want to detect cytochrome C, if you look at it in literature, what people do is they use UV visible absorption spectroscopy, right? So in that, what you have, this is the typical UV vis absorption graph of cytochrome C. So the horizontal axis is the wavelength and the vertical axis is the absorbance, right? So now cytochrome C is a heme molecule. So iron can be in two states. It can be in either Fe2 plus or in Fe3 plus, right? So now if cytochrome C is in the reduced state, you will have two peaks around 550 nanometers shown in blue. However, if cytochrome C is in the oxidized state, you will have a single peak around 520 nanometers as shown in red, right? Now, since it's a redox reaction, you can always reduce it and oxidize it. Now, for reducing it, you add a reducing agent like ascorbic acid, and for oxidizing, you add a chemical like hydrogen peroxide. Now, this is typically what you can do. This horizontal axis is the time, and vertical axis is the absorption at 550 nanometers. So higher the absorption, more the reduced cytochrome C that you have. So at till this point, we did not add any chemical. At this point, we're adding ascorbic acid. So the absorption goes up, so it becomes more and more reduced. And at this point, we are reducing it, we are oxidizing it. So it reduces in intensity, right? So this is typically what you can do. However, if you notice, the concentration that I used here is 10 millimolar, which is very large, right? So now on a on a single particle, we have around 100 molecules. So that would come in femtomolars or zeptomolars, right? A very small concentration. So UV visible absorption cannot work with single particle measurements. And in fact, if you take the synthesized particles and try to do UV vis, you will not get a signal, right? So we have to enhance the signal somehow. And that is where plasmonics comes in. Now, the simple system that you can do is you take these particles that we have synthesized. Now you basically make an aggregate by reducing or increasing the pH. Now, once you have these aggregates, between all these particles, you have the junctions which have a very small gap. Now, if you remember that if you reduce the gap, you have a high field. The same principle applies here. Right? So all these junctions will give you a high signal for Raman. And that is precisely what we use. So for this structure, if you send light from the top, the laser from the top, and look at the reflection, what we get is shown in this plot here. So the color map here, the horizontal axis is the wave number or the Raman shift, and the vertical axis is the time. Now Raman, we know very well that it always fluctuates because the molecules are in water and the molecules keep moving, right? So if I notice, if I take two different times, like the red curve and the black curve, and look at the cut that is shown at the bottom here, what I see is I see nice peaks which correspond to the Raman peaks from the molecules that are there in the sample, but they vary significantly. So the red one is very different from the black one. Again, this is amplified by the fact if you look at a single wave number, in this case 1590, and as a function of time that is shown here. And you see the intensity fluctuates significantly. So what do we do? What we do is we do a statistical analysis. So we look at a parameter called as the Q parameter, which takes the variation of these peaks into account, and then we can plot that. And that is shown here. So from this whole data as a function of time, we can reduce it to a single spectrum that shows looks like this, which takes all the variations into account. And then we can see clearly all these peaks that we have in the Raman spectrum, and then we can use it for analysis. Or we can look at the histograms of the peaks and still even use that for different analysis, right? Now, the question that still arises is that, look, we are doing it for arrays, for big aggregates, which has thousands of particles. Can we come down to a single particle and do the same measurement or measure the Raman from a single molecule, right? So the idea then comes is you use something called as a plasmonic antenna. So I'm sure every one of you has seen a tweezer, right? Or a forcer. Now in a tweezer or a forcer, what you do basically is use that to catch and hold small objects. Now these gold antennas or the gold cuboids that I showed before also act like tweezers, but at the nanoscale. So here we have two different gold bars shown by the yellow cuboids, which basically form a plasmonic tweezer. Now, if you shine the light onto this structure, what will happen is you will have a high field which will actually try to get the particles from outside the trap to inside the trap and get them between these two cuboids in the center of it, right? So this is the experiment that we did. So again, the horizontal axis is the wave number, the vertical axis is the time. So now the strength of the tweezer or how hard you are holding it depends on how much intensity of the light that you're sending to this particular structure, right? So here what we are doing is initially the laser is off, then we turn it on at this point at a low power, but we still don't have any trapping. However, at this point, we actually increase the power so that it's enough to actually hold these single particles. And what we see is after a certain time, after we turn on the laser power, increase the laser power, we see that, look, we start seeing this Raman peaks that we start appearing in this spectrum. Again, after a certain time, when we think we have enough spectrum, we stop the laser and we let the particle move out. And then if you turn it on again, we don't see the particle anymore, right? So what this tells us is here there was no particle between 0 and let's say around 100. Between 100 and 120 in seconds in time, we have the particle being trapped and we can measure the Raman. And then again, we release the particle. So this is a typical example of plasmonic tweezers. And then this is the spectrum that you see in the trapped region and you see very nice peaks, right? Now, if you actually calculate a rough estimate of the number of molecules, you will get around 100 to 1000 molecules on each of these plasmonic particles, right? So what you're basically going down, you're reducing the amount of molecules from millions of molecules to barely 100 or 1000 molecules on a single particle, right? And then, of course, you can compare the peaks that you have. For example, here, these are the different Raman peaks that we see for the case of the aggregate and the trap. And then we see that most of them match. And then we are able to conclude that you have cytochrome C in both the samples. However, for the trap particle case, we have very few molecules from which we are getting the signal, right? So now, can we do some kind of diagnosis with this? Now, the way that the diagnosis works is we exploit the property of these cytochrome C molecule that we have on those particles, right? Now, cytochrome C is a very interesting molecule because, as I said, it has this redox reaction kind of feature. Now, one way of oxidizing it is using hydrogen peroxide. Now, hydrogen peroxide is a chemical that also is produced in the body when you have a lot of stress, right? So whenever you have stress, the level of hydrogen peroxide or reactive oxygen species actually goes up. So by measuring how much of reduced and oxidized cytochrome C you have, you will be able to tell whether you are in a stressed condition or not. So this is a typical Raman peaks that you get. So horizontal axis is the wave number, vertical axis is the Q parameter. And if I zoom into this certain part of this spectrum, what I see is if I have the oxidized one, I'll see 2 peaks. If I have the reduced one, I will see 1 peak. So by looking at this change, I'm able to say whether the cytochrome C on my particle are oxidized or reduced. So this is one way of doing diagnostics with SIRS. Now, if you notice here, I said we need high laser powers. Now, we know also one more thing, that biological objects like cells and tissues are very prone to damage based on heat, right? So this technique is sometimes difficult to apply to cells and biological tissues. So the question is, can we also analyze these particles using a different technique? Of course, the answer is yes. There are multiple ways of doing it. One more way using plasmonics is using these nanohole apertures. So what we have is basically the silicon nitride membranes, which is shown in the gray layer, and a layer of gold on top, which will explain to you how we fabricate it. And then the idea is we want to pass these nanoparticles through these holes and measure how the signal changes. So the way we make these structures, we have a multi-step lithography process. So we start with a silicon wafer coated with silicon nitride on both sides. We coat the top part with the photoresist and then do a layer of, we do a step of photolithography to make the apertures that we want. Then we do the etching of silicon nitride and then do the etching of silicon using KOH. Now, KOH etching of silicon 100 leads to the formation of basically what you call a pyramid kind of shape that is shown here. So this is the actual fabricated structure that we have. So this is the pyramid shape, and this at the bottom is the thin membrane of silicon nitride that we have. Now, once we have this, we deposit a layer of gold and then we do focused ion beam milling. Now, if you have done FIB or you have studied FIB, you know that look, if you have soft materials like gold, you will mill them off at a very fast rate. So before you can actually make through holes, you will have the gold getting removed. So this is a typical example of when you do milling without any protection, any hard layer on top or a hard mask at top. So if you notice here, the walls are not very flat. And if you make very small structure, these are 50 nanometer holes. So you see that you get hills rather than flat walls. However, if you put a layer of chromium, you get walls which are very, very flat and sharp. And these are the typical holes that we have fabricated for this particular structure. Now, if you do the experiment, so the experiment is very simple. So you take the silicon nitride membrane, which is supported by this the substrate. You put basically a well of water at the bottom and a container which contains the gold particles at the top. And then naturally the gold particles will diffuse from the top to the bottom based on diffusion. And then we would like to measure when these particles are diffusing. So, and then you send light from the bottom and measure it in reflection to see the signal. Now, if both of the chambers are filled with water, if you measure as a function of time, you will see no signature or no signal change. It will be practically zero. However, if you actually put these nanoparticles in the top chamber, what you will have is you will see signals like this. So till some point, the signal will be zero. After some point, you will see some modulation. Now, this modulation is what comes from these nanoparticles. You can actually simulate it and simulate it by putting these nanoparticles at different positions in the channel. And then you'll get a similar response as compared to the experimental result that we have. So what we're able to do here is we're able to basically measure the particle as it is going through this single channel that we have. Right now, you might ask me, how is this connected to SIRS? Now, if you look at the calculation, these are shown here. if you place this particle, that is this center circular region that you have, in the hole that is shown by the outer green circle, if you look at the fields, it is very enhanced. So it is enhanced by a factor of 15 as compared to when you don't have a particle, right? Now this is the cross-section view, and there are two different particle sizes. So what we see is there's a high field between the particles wall and the wall of the cylindrical hole that we have. So if we place molecules here, the Raman will be significantly enhanced. So basically 15 will be enhanced by a factor of 15 to the power 4, right? So this is what we plan to use for doing search using these structures, right? So the idea here is, this will be the first part. I would like to conclude the first part. So what I would like you to understand is that, look, plasmonics can be used for studying the signatures of molecules, especially biological molecules like proteins, at a single particle level, which have very few molecules on these at this concentration values, right? Now, the future of this is, of course, that can we study more complex geometries of these molecular layers. Let's say if you have 5 or 10 different proteins, can we analyze what are these proteins and what is their concentration? But that is still a work in progress. Now, let me come to the second part of my talk, which is based on distinguishing between normal and cancerous cells using surface enhanced IR absorption spectroscopy, right? Now, this is the analog of search in the mid-IR spectral range, right? So now you might ask me, like, why is this spectral range important, the mid-IR spectral range? Now, like in Raman, which is based on scattering, scattering, you also have vibrational fingerprints in the mid-IR spectral range, right? And typical nomenclature in the mid-IR spectral range, you talk about wavelength in centimeter inverse, not in nanometers or centimeters, right? So this is 1 by wavelength. Now, since they are fingerprint bands, by looking at where these bands are occurring, you can actually say what these molecules are. Now, if I give you two examples, if you look at the amide bond, CONH2 bond, right? This has multiple vibrational features. So some of them are the amide one, which occurs typically between around 1650. Then you have the amide 2, which occurs around 1540, right? So if these peaks are present, you can say that, look, you have a molecule which has amide bonds. Similarly, you can say, look at the phosphate bond or the carbon-carbon bond or the bonds corresponding to OH or so on. However, if you have done FTIR or look at the FTIR setup and the sample preparation, you know that, look, you need a very thick sample or a high concentration sample. If your concentration is very low, you will not get a very nice signal. So that is a perennial problem with IR absorption signals. So again, we go back to the same idea that, look, plasmonic structures can enhance our signal. So we'll use plasmonic structures to enhance our IR signal from molecules. So these are the typical metasurfaces I have shown before that we use for these studies. So we have a single cuboid, which is a linear bar with an L-shaped bar, right? Now, if I send light from the top and look at the reflected light, I will get a response as shown here. So this is the wave number, 1 by wavelength, and the vertical axis is the reflectivity. So I'll have multiple features. So let us see what these features are. So this peak here and this peak here correspond to the two plasmon resonances of my structure. Now, the way we typically say it's a plasmon resonance is by looking at the near field. So if you remember, at plasmon resonance, your field very close to the structure or in the near field is very significantly enhanced, right? And that is the case for both of these wavelengths, right, as shown here. So the field is enhanced as close to this straight bar and the L-shaped bar at both these wave numbers, right? Now this, however, since this is a resonance structure, what we have is, as we go away from resonance, the field intensity decreases. So if I take a wavelength that is in the center here, the intensity is not as high. Okay. So typically, if I have some molecular band here and I want to enhance it, I would like to put a resonance that is at that particular molecular band. So the way that we work to solve this problem is we make structures which are tuned to different molecular features. So the simple way of tuning is that you take this unit cell, which has a period of lambda x and lambda y, and then you scale it so that you increase the size of all the dimensions that you have. Now by scaling, so if I go from a smaller size to a larger size, my resonance will move from around 2500 centimeter inverse to around 1500 centimeter inverse, right? And this is a typical optical image of the samples that we are fabricating. These are the smallest arrays and these are the largest ones, right? So if I put different structures, I'll get different enhancement for different bands. Now, since we are interested in looking at how cells are responding to these structures, we also need to look at how deep we are actually probing into the sample, right? What is the depth that we are actually getting the signal from? So this is a calculation where we are plotting the field in the XY plane, which is this horizontal cut, and the Z axis, which is in the vertical direction. Now, if you notice, this is around 2 microns in height. So if you notice this region, which is shown in red and green, it is very, very small as compared to this. And this region is around 60 to 100 nanometers from the surface of the structure. So what this is telling us is we are only enhancing the molecules that are that close to the structure, only between 60 to 100 nanometers. Now, if you compare it with cells, now cells have a typical thickness of 1 to 4 microns. So what you're actually measuring is not the whole cell, you have to remember. You're measuring only something which is very, very close to the cell membrane, right? And that is precisely what we use for the diagnostic purpose. Now, you can also verify this experimentally. So here what we are doing is we are using two different metasurfaces with two different sizes. The horizontal axis is the thickness of the PMMA layer, which is the polymer on this metasurface. And the vertical axis is the signal that we are measuring. What we see is as we increase the thickness of PMMA, beyond around 100 and 150 nanometers, we don't see a large change in the signal. So what this tells us is even if we make the thickness of PMMA, let's say like 500 nanometers, the signal will again still be around 0.35 microns, right? It will not change significantly. And that tells us the typical dimension that we are actually probing, okay? Now, we started with simple experiments. So what we initially started with to check whether the system works properly is can we distinguish between different solvents, right? So we took three different solvents. One is PBS, which is basically a buffer that we use for cell culture. So phosphate buffered saline. And we chose two different solvents like ethanol and acetone. And the system that we use is basically a microfluidic channel where we have the metasurface that is encapsulated in this channel so that we are able to flow liquids and also measure in static condition. Now, if I take three different cases, when this chamber is filled with either PBS or with acetone or with ethanol, we see, if you look at the IR spectrum, the reflectivity versus wave number, we see three different spectra, and each of these bands that we see, the sharp features that we see in the red and the blue one and the black one, can be identified with the vibrations that you have in these molecules that we are looking at, right? Now, you're not limited to doing static measurement. You can actually do dynamic measurement that is shown here. So we start with the chamber when it is filled with PBS, which is close to water. And then we flow ethanol after some time. We start injecting ethanol. So it will take some time for the ethanol to completely replace water or PBS in the chamber, right? During this whole process, you can measure the signal and you can basically get a plot like this. So horizontal axis is wave number, vertical axis is the time, and the color represents the signal that you're measuring, right? The reference here is of course the initial state of water. Now, if you plot as a function of time at this one of these bands, we see that the signature of water actually reduces and the signature of ethanol actually increases. So you can do dynamic measurements using this system, right? Then if you are just interested in sensing, you can make a calibration chart. You do a series of concentrations starting from pure PBS or a mixture of PBS and ethanol at different values. And then you see that the signal actually changes progressively. And by making a calibration curve, you can say how much of ethanol is there in the solution, right? Now let us come and apply this system to actually to human cells and see how we can apply it for diagnostic purposes. So what we do is this experiment was done in multiple steps. And in particular, we use two different kinds of cells. We use colon normal cells and colon cancer cells, right? So we start with the metasurface, which is in buffer PBS. Then we put a layer of antibodies, which are basically used for capturing the cells that we have that we'll put in the chamber. Then we allow the cells to actually get captured. So this is a typical image of the cells, bright field image of the cells. So each of these blobs is a cell, and the dark gray region that you see is basically the metasurface. You see around 10 to 15 cells on these metasurfaces. Then you basically let the cells grow for 4 hours so that they'll spread out. So if you see these cells as compared to this, you will see that they have this philopodia and they are spreading out. And then finally, you can fix the cells to look at them at a later time, right? So we measure the Raman, the IR signature at all these time steps, and then we can plot them in a single plot as shown here. So the vertical axis is reflectivity, and the horizontal axis is wave number. Now, if you notice, there's not a major change, right? However, if you actually look at either the derivative or the absorbance, which is typically done in IR spectroscopy, we start seeing that, look, for different states, we start seeing different features that start arising, right? And these are the features that we can use for identifying different kinds of cells. So, but what you have to notice is this features, in some cases, will go higher, some cases will be lower. So we cannot do it manually. So we do something called as principal component analysis, right? And this PCA basically allows us to identify very complex data set and it tells us whether there will be a significant difference between these two data sets or not. And in this case, it's two different spectra, right? So here what we are plotting is the PCA scores, principal component scores, between multiple sets of data. So the horizontal axis is the principal component 1 and the vertical axis is principal component 2. Now you see different colors. Each of the colors arises from a different kind of cell. So the red one corresponds to the normal cells. The blue and the magenta ones correspond to two different kinds of cancer cells. What we see is they form three different clusters, right? So what this is telling us is by looking at where these points are actually falling, we are actually will be able to tell whether these are cancerous cells or normal cells. And that's the whole point. So this was something called as unsupervised learning, right? So we did not know what each of these, we knew what exactly each of these categories were. What we are working on currently is the supervised learning, where we compare a given spectrum to the already known set of data, right? So as I mentioned earlier, now this can be used to apply it for understanding the differences and identifying cancerous versus normal cells. Now the advantage is we need very, very few cells. And the other major advantage is we don't need any fluorescent labels or markers, right? Which most of the other techniques require that are used in diagnosis, right? So now the question, however, you might ask me, which normally will come up, is that look, these cells we are doing it in all these multiple steps, but this is a crucial thing, right? We need to get the cells to this metasurface. How can we do that? Because if we do it the way that we have done in the exploratory experiment, it will not be practically possible to do it in the field, right? So how do we do it and actually get the cells from somewhere in the bulk liquid to the surface? For that, we used one technique called as dielectrophoresis, or you can use other techniques. But in our case, what we used was dielectrophoresis. And what that does is you have a metasurface, you apply some kind of AC field, electric field, low frequency electric field, and this low frequency electric field will actually get these cells that you have to the surface. And then you can do the IR measurement that you would like. And then you can do the classification using principal component. Right? Now, the idea here is what we envision is that, look, we don't want to use cells in a nice liquid like PPS or buffer, but we actually would like to flow blood or some body fluid, capture the cells using this geometry, and then measure the signal from these captured cells, right? So this is a geometry that we actually fabricated and used. So what we have is wires, interdigitated kind of wires, right? So one connected to the positive electrode, the other connected to the negative electrode, and this is a typical SEM image. So we have the same structures as before, and these are the wires that we use as electrodes, right? Now, if we send light and look at the reflected light as a function of wave number that is shown here, we again see a similar feature that we have a resonance here and a resonance here, right? Now, if you actually simulate the response of the electric field and calculate the forces, which is what we did before we did the experiment, this you can do using COMSOL, right, as shown here. So this is the typical channel that we have, and the color actually here shows you the flow velocity. We have a laminar flow because the Reynolds number is very low, right? And if you look at the cross section that is shown here, so the color here represents the DEP force. So if you have a blackish purplish color, you have very high force. However, if you have a white or a yellowish background, the forces are very small. And the lines that you see here correspond to the trajectories that are followed by the particles, right? Or the cells. Now, these are where the electrodes have been placed. And around the electrodes is where you have the metasurface. So if I have a cell which comes and sticks here, it will basically be sitting on the electrode and on the metasurface, which is what we'll use. So this is a typical simulation that we get. So there are multiple electrodes that we have on the same device. And at each of these electrode interfaces, we'll have a high particle density. So the particles will come and stick onto where the electrodes are, right? So this is the experiment that we have done, right? Again, you see a very similar variation that you'll have maximum density of particles at the edges where the electrodes are, right? So this is a typical, what you call, experimental image that we get. So here we have different kinds of particles. This is a polystyrene particle which is fluorescent, and these are two cases of different kinds of cells. Now, if you look at the dielectrophoretic force, which is given by this equation, you will have the gradient of the electric field, which is why the field is maximum and the force is maximum near the electrodes. You will also have a term which is dependent on this KRE. Now, this KRE is dependent on the frequency omega and the type of cell that you have, right? So different cells will experience different forces at a given frequency, right? So by choosing the frequency, you are able to attract some cells and repel some cells, right? That's the whole idea of dielectrophoresis. So what we are doing here is, when we apply the AC field to this electrodes as a function of time, we start seeing that, look, the cells will start accumulating along these electrodes, which is shown by the white lines that we have, right? So you increase the time, you start accumulating more and more cells. This is the same case for all the different, three different cases that we studied of the two different kinds of cancer cell and the polystyrene particles that we used as a reference for our study, right? So now, just in addition to just capturing the cells, you can also measure the spectrum as these cells are getting captured, right? So this is a typical graph that we get. So horizontal axis is the wave number near the amide 1 and amide 2 features. This is amide 1 and this is the amide 2 feature, and this is the absorbance that we get, right? Log of I by I naught. Now, as I increase the time, so more and more cells are getting captured, I see the signal getting progressively increases. Now, corresponding to this particular experiment, you can see this is the actual image that we get of during the experiment, and we see that, look, the place from where we measure the signal, the density is actually increasing, right? And from this particular set of images, we can calculate the coverage, and from this we can calculate the signal, right? Which we use the area under the curve. Now that is shown here. So this is time, and we see the coverage and the area are exactly following the same trend. So what this is telling us is that the signal actually comes from the cells that are getting adhered or getting trapped because of this dielectrophoretic force on these particular metasurfaces that we have, right? The same thing can be done with this A431 cells, which are lung carcinoma cells, and then you see a similar trend, right? So what we show here, and what I would like you to understand and take back from this particular slide, is you can do simultaneously the capturing and measurement of these cells that we have in a fluid, right? We don't need the antibodies for doing all this capturing studies. Now, another place where we can think of applying this metasurface is the following. Now, if you are familiar with cancer therapy, right? typically doctors go with literature study and they try different drugs at different stages. However, a lot of times doctors do combination of drugs so that there's better chance of treatment for that particular patient, right? This is basically a kind of a guesswork that the doctors do, right? So what people are trying to do now is it possible to actually predict for a given patient what kind of drug combinations will work the best for a given kind of cancer and given stage of cancer, of course, right? So we tried to do a similar study. So in this case, what we did was we take primary cells, cells which are actually taken from an animal, in this case from feline, right, which had different stages of cancer. Now we treat these cells with different drugs. Now we either treat it with doxorubicin, which is a standard anti-cancer drug, or salinomycin. There are two different drugs that are used, or a combination of them for a certain time, which in this case is 48 hours. Now, as a reference, we also take a case where the cells have not been treated. Now, the question that we have, or we asked ourselves, is can we use this metasurface based diagnosis for basically identifying what kind of treatments, whether the treatment is working or not, and whether it's effective or not, right? So we do the same kind of experiment as before, that and measure the IR spectrum when the cells are put on the metasurface, and then do the principal component analysis. Now, if you notice, this is a principal component plot between PC1 and PC2, and we see 4 different clustering. So if you remember, for the 4 different colors correspond to the different case of drug treatment, right? So what we see clearly is by changing the drugs that you're using, you see actually a major change in the principal component score. So what this is telling us, yes, we see a difference based on the treatment. So what we are working on right now is can this treatment, can this clustering be associated with the efficacy of the drugs? That is something that is still a work in progress. Now, another thing that you can think of in terms of metasurface is, look, why do you really want to make metasurfaces which have lots of different resonances? Can we just use one metasurface with one single size and somehow tune the resonance so that I can use it for sensing, right? So, which is a very simple thing to do if you actually include some material which will actually allow you to tune the resonance, right? So you really want to start with this kind of structures where you have metasurfaces as shown before, which have the same resonance, resonance. So you have to change the size. Now, if I actually integrate a layer of graphene, now graphene has very interesting properties, you might all know, and people have used it for various applications, starting from battery to sensors to what you call for lubricants and things like that. In this particular study, we are using graphene for changing the carrier density and thereby controlling the resonance of our structure, right? So graphene, as we know, is a is sub-nanometer thick layer of sp2 hybridized carbon, right? It has excellent electrical properties, and you can tune these electrical properties by using gating or by making a field effect transistor, right? So the way that we work with these devices is we have a layer of metasurface which is placed on this single layer of graphene. So remember, a single layer of graphene, not multiple layers, which is placed on a layer of SiO2 and a silicon substrate. Right. Now, if you connect these 2 electrodes, which is placed on the graphene and the silicon, you can actually apply a field. Right. Now, by controlling this field, you will be able to control the Fermi level of graphene and thereby control its absorption. Right. So now, if you look at the send light at normal incidence and look at the reflected light, right? So here, what we are doing is we are looking at the reflected light as a function of the gate voltage. So the voltage that we are applying between the drain, that is, or the drain electrode, and the back gate, which is the silicon. Now, this is the simulation. So horizontal axis is the wavelength of light that we are sending in. Vertical axis is the reflectivity. Now, as I increase the Fermi level from 0.08 electron volts to around 0.23 electron volts, I see that the resonance wavelength, which is shown by this dip, which is around 7.8 microns per 0.08 electron volt EV, will actually move to around 7.6 microns, right? So which is shown in the purple curve. So what this is telling us, and similarly in experiments, of course in experiment we actually change the voltage. So this is shown in the voltage that we're applying between 0 and 320 volts. What we see is the resonance changes from 7.8 microns to around 7.6 microns. So what this is telling us is we can actually change the resonance and thereby choose which molecular feature or molecular absorption band this resonance overlaps with, and thereby enhances, right? So this basically allows us to reduce our structures from multiple structures that we have, which we basically made because our resonance was limited to one particular molecular feature, and we can use this tunable structures for sensing different molecular absorption bands, right? Now the question is, of course, if you want to do sensing, you really want to do it in a tunable fashion, right? You do it in a sequential fashion. You do amide 1, then amide 2, then amide 3, then come back to amide 1, right? So the question arises is how fast do you do it? Now for that, we do a sweep in time, right? And we keep increasing this frequency of the gate voltage, omega g, and we look at how fast the modulation can actually occur in this device, right? And that is shown here. So here we change the frequency of the gate voltage from, let's say, around 10 to the power 4 hertz, 10,000 hertz, to around 10 to the power 7 hertz, right? Which is 3 orders of magnitude. And what we see is during this whole range, which was limited by our instrument that we have, we see that during the whole range, we can actually get the same kind of delta R, which is the signal. What this is telling us is even at these high frequencies of 10 to the power 7 hertz, we can still get the same response. So the device is responding normally. However, if you actually increase it further, go to 10 to the power 8 hertz, which we have done later, we see that the device cannot respond anymore and the signal actually reduces, right? So what you can actually do with these graphene integrated devices, as I have shown here, is that look, you can reduce the multiple pixels into a single pixel device, right? Which will make it much easier for a practical device application, right? Now, of course, there'll be one more thing that you can ask, that look, initially I started the presentation by saying single particle source. Now, can we do single particle IR absorption, right? So we wanted to study that. So one of my students at IIT Hyderabad was working on that project. So what she did was basically she takes this kind of plasmonic antennas. So each of these bars A and B, labeled A and B, are made up of gold. And then in between these two A bars, we have a small gap of around 100 nanometers. Now, this is a typical meshing that we use for doing the numerical simulations. Now, if you look at the response of this structure without this particle, we see something like this. So where the horizontal axis is the wavelength and the vertical axis is the reflectivity, right? So now, if you notice, we have multiple features again. So we have two peaks and one dip. So the resonance of the structure is at this, near this peak and near this peak, right? Now, as we know, if we have high fields, we can also use those high fields for capturing objects. So that is shown here. So if you notice, between these two bars, we have a very high field. And if you actually plot it, it is maximum near that resonance that I was talking about, right? So, and the force that we have or we apply on the object is proportional to the gradient of the field or gradient of the intensity. So that will be, the objects will get trapped in this particular gap here. So this is a simulation of that. So we are putting a 50 nanometer particle in this 100 nanometer gap and looking at the different forces, different changes in reflectivity we'll have, and the field that we'll have on the particle, right? So if I take the structure with and without particles, the response is shown here. So the horizontal axis is the wavelength and the vertical axis is the reflectivity. So if you notice, when I place the particles of two different sizes, 20 nanometers and 25 nanometers in radius, there'll be a small shift of the resonance. If I zoom into this region, I will see the shift much more clearly, right? Furthermore, if I look at the field, if I calculate the field on this particle that is trapped in this gap, what I notice is that the field is enhanced by a factor of around 35, right? So what this is telling me is the IR absorption signal will be enhanced by a factor of 35 squared, right? which is around 900 times higher as compared to without these structures, right? So that is what we were interested in doing. And so this tells us that, look, you can actually apply the concept that we had used before, that of trapping and sensing, even in the case of IR absorption. It is not just limited to the case of Raman scattering. So with this, I would like to start concluding my talk. So what I hope what you got from this is nanomaterials, in particular, plasmonic nanostructures and metasurfaces are a very fascinating area that have a lot of applications. You can use it for diagnostics, for devices. You can do interesting things which increases the potential of various applications that you can think of, right? In addition to that, you can also think of applying these structures for getting signals from very small volumes and concentrations of molecules, right? So briefly, I will go over what we do in our lab. So at IIT Hyderabad, we work on plasmonics in different aspects of plasmonics. So we work with the materials, we work with nanofabrication, we work on the characterization, the modeling, and then of course, all our applications, all our work is of course directed towards application. Currently, we are looking at sensing and optical modulators. And these are typical images of structures that we have fabricated over the course of time. So these are silica particles, which have been which have been made using Strober's process. These are structures. So each of these dots that you see, the bright regions, cylinders, are gold cylinders with a diameter of around 80 nanometers and a gap of around 20 nanometers. So this was made using electron beam lithography. This is basically a plasmonic metasurface. And these are plasmonic metasurfaces, but much larger in size, right? So now the latest that I would like to briefly show is basically, look, till now we have been talking about plasmonic structures on a flat substrate, right? Let's say a piece of glass or a piece of silicon, which is basically macroscopic in size, right? You have a 1 centimeter by 1 centimeter kind of size on which we typically make the structure. Now the question is, can we do something better? Well, yes, you can. So for example, here we have an optical fiber which works in the mid IR. And on top of that, we have actually used a technique called as focused ion beam milling, right, to make nanostructures. So if I zoom in, you start seeing these bars which are being made on top of this structure, right? Now these bars are made up of gold. So now this will allow us to do different kinds of things like kind of make chemical endoscope. So think of this device or this fiber. Now you can get this fiber in contact with the tissue and then get the signal, and then you use it for different analysis, like how we did for distinguishing between cancer and non-cancerous cells, right? So this is a very interesting piece of area which we are actually actively pursuing, right? So this is the lab website, which is, you can find it at this link, ith.ac.in/Shaurya. And so please feel free to send me an email if you find something interesting, or you would like to work with us, or you would like to visit. We are always happy to have new people joining our lab and visiting IIT Hyderabad and our lab, right? Now, so in conclusion, I hope you can take these 2 points home. So one, plasmonic structures have a lot of potential, right? Now you can use these plasmonic structures for enhancing the Raman intensity of molecules, right? Now the intensity is proportional to the electric field to the power 4. And by choosing the structures properly, you can enhance the Raman significantly by a factor of 10 to the power 6, 10 to the power 7 is normally what you get, right? And by using this technique, you can actually measure the Raman signatures of few hundreds of molecules on a single particle level, right? You're not looking at millions of particles, you're looking at a single nanoparticle and analyzing that. Now, the other part, I hope that I could convince you that, look, the surface enhanced IR absorption, or SIRA, right, as we call it in the community, can and has a strong potential for cancer diagnosis. And the reason is very simple. I will leave you with this thought. Look, cancer is a disease which progresses from stage 1 to stage 4, right? Now, if a patient is detected in stage 1, he has a much higher chance of remission and for improved lifestyle as compared to stage 4. So you always want to detect the cancer in stage 1 or stage 0, that's even better as compared to stage 4. So you really need to detect the cancer when you have very, very few cancer cells. And as I showed that here in this particular technique, you can detect the cancer cells using just a few cells. Even 2 to 3 cells are enough to actually do this diagnosis. And then, of course, I showed you a technique which can actually help you to capture these cells and get them to the metasurface, right? So with that, I will leave you with these thoughts. So this is the photo of IIT Hyderabad. These are 3 principal buildings that we have and the different labs that we typically use. So we have the optics lab, the clean room and the fabrication facility, and the sputtering lab that we use for making all these devices. Thank you.
Thank you very much, sir. It's a kind of a super journey. Actually, it was a fantastic lecture. Now I request Shainita to read some of the questions. And also, in the meantime, Shivagaru, some of the participants, if you want to talk, we can give permission to them. So, dear participants, if you want to talk or if you want to ask, interact with Dr. Shaurya Dattagupta, you are most welcome. Please raise your hand so that the technician will give you permission to speak here. Okay. Now over to Shainita.
One of our participant has got a question, sir, but I hope he did not clearly type it. But let me read it. Sir, you have mentioned about nanoparticles with corona when find out or calculate the solubility and dissolution study of nanoparticles. Did you get my question, sir?
Can you repeat it once?
Actually, the question is not clearly typed, sir, but still I once again see. He's asking about like did you mention about the nanoparticles with corona? When? Then how to find out or calculate the solubility and dissolution study of these nanoparticles?
Okay, so if I understand correctly, you want to study how soluble these particles are, right? So if you might correct me if I'm not getting the question, but the basic thing is it depends on the kind of protein that you're actually using, right? So now if you have a very hydrophobic protein, right, which has a lot of hydrophobic groups, that is not, or rather, let me put it, if you have a lot of lipid molecules in your particle, they will not be very soluble in water. So it's a competition between the molecules that you have. Now, you are typically making a protein corona with different kinds of molecules, and you put them in water. The hydrophobic molecules typically go towards the inside. So they are screened by hydrophilic molecules, and you have a minimization of the energy of the particle.
Our next question is SERS technique available in diagnostic centers?
So SERS is something that people are actually actively making devices and what you call instruments for diagnosis. For example, RR is Raja Ramana Center for Advanced Technologies. It's in Indore. They are making devices based on SERS and for diagnosis of cancer. They are actually testing it with patients in Tata Memorial Hospital in Bombay. Okay, so this is something that people are actively pursuing. However, there's still a lot of challenges that need to be solved. For example, how small a sample do you need? How many few cells can you measure? And what is the condition of the sample that you need it in, right? So this is a very active area and you have a lot of potential for making instruments and devices for the market.
If we are capping the nanoparticles protein to produce drug, then how to calculate the solubility and dissolution study? Did you get my question, sir?
Yeah.
I'm asking if we are capping the nanoparticles with protein to produce drug, then how to calculate the solubility and dissolution study?
So we did not do the dissolution study, so I'm a little hesitant in answering. But typically, these what you call so first of all, this cytochrome C that we have, right, on each particle we can actually nominally calculate how many molecules we have based on the size. Okay, now if you actually want to calculate how many molecules you have, you can actually dissolve the molecules and do a quantitative mass spec to get the number, right? So these are the only two techniques I can think of to study the solubility and the amount of, let's say, drug that you have on the molecules, on the particles.
Yeah, generally pharmacy people will be doing the solubility and dissolution studies mainly.
Yeah.
Shanita, next question you can ask.
Yes, sir. Is
Shanita, one more request. After asking the question, please mute the mic so that it will be audible to all the participants. Okay?
Our next question is IR radiation used only for, is IR radiation can only be used for diagnosis of cancer, or it can also be used in cancer cells?
Can you finish the last part?
I repeat the question, sir. Is IR radiation can be only used for diagnosis of cancer, or it can also be used to incinerate cancer cells?
Okay, so that's a very interesting question. So the question is basically, can we use IR radiation to kill cancer cells, right? So indeed it is possible, and that is called as photothermal therapy of cancer using plasmonic nanoparticles. There's a big group in Rice University which has pioneered this area, right, by the group of Naomi Hallis. So the idea is that you take gold particles and you send IR light, in this case particularly near IR light, not mid IR light, and now these particles will get heated up, right? And this heat will actually kill the cancer cells, and that is what they use for photothermal therapy of cancer. So yes, you can use it and for therapy.
Sir, one of our participants is working on pegylated nanoparticles for treating Parkinson's disease and cancer. So he's asking, like, is there any PhD research going on this topic, so that he wants to join?
So currently, I have a few students who are working on biosensing using plasmonic structures using SIRS, right? However, as part of our work, we routinely work with chemicals like PEG, polyethylene glycol, or thiolated PEGs for doing different kinds of functionalization, right? If he's interested, he is most welcome to contact me and we can talk about his interests and the possibilities.
Thank you, sir, for answering all the questions. We are done with the questions.
Shiva Garu,
Yes, sir.
can you unmute Shanti Ganapa? They raised her hand. They want to talk with sir once.
Yes, sir. I have unmuted her, but there is no response from her.
Shanti
Hello.
Ganapa. Yeah.
Hello.
Yes.
Yes, sir. Sir,
Please introduce yourself and talk, Shanti Ganapa.
yes, sir. Hello. Good afternoon, sir.
Yes.
So recently, I'm pursuing my post-graduation in the nanotechnology, sir. Final semester. Due to this COVID-19, all got stuck, sir. But I'm working on the pegylated nanoparticles, sir. So is there any research is going on under you, sir?
So as I said, I don't make PEG particles, but we use PEG for different functionalization. So as you know, PEG is typically a very good anti-fouling material. Okay, so
Yes.
that doesn't allow other proteins to come and stick. So we use that routinely
Yes.
for making devices which are anti-fouling in nature. Okay, so
Yes, sir. Yes, sir.
our interest is more the plasmonic thing. So we will always typically use either gold or silver kind of particles for doing this kind of devices, right? So, but
Yes.
if you
Yes,
are
sir.
interested in, if you can think of a way of using PEG particles in sensing, which people have done, I would be very happy to talk to you. I mean, that's of course an area that is interesting to us.
Yeah. Sure, sir. Sure, sir. So can you share your details where
So
I can
you
contact you, sir?
can visit this website, iith.ac.in/shaurya, right? And Professor Venkateshwar Rao can, I think he has put it already on the brochure. So you can just send me an email about whatever details
Yeah.
you need.
Sure, sir. Thank you, sir.
Yeah. I think Shainita, I think questions from YouTube also. Did you selected the questions, Shainita?
Yes, sir.
From YouTube also some questions are there. Did you complete that questions also?
Actually, there are no questions from YouTube, sir. I have seen.
Checked it? Did you check it?
Yes, sir.
Okay, then no worries. So thank you very much, sir. It was a fantastic talk, and happy to interact with you all. Interact with you, sir, and happy to listen about sirs and all other things. And thank you, sir. Thank you very much.
Thank you, Venkateshwara, Professor Venkateshwara. Thank you.
Thank you. So in the meantime, just could you please share the Saurya Datta Gupta sir's feedback link, Shainita, in the group?
Yes, sir. I have shared it, sir.
Yeah, already Dr. Bharati Mohan came to online. So Shiva Garu, could you please make Bharati Mohan as one of the presenter?
Yes, sir. Done, sir.
Done. Dr. Bharati, could you please share your screen? Yeah. Good morning, Dr. Bharati Mohan. Hello. Good morning, Dr. Bharati Mohan. Very good morning. Yeah, your voice is little low actually. And could you please share your screen? Yeah. Yes, Bharati. Last screen is there. Yeah, fantastic. So, a very good morning to Dr. Bharati Mohan from Pondicherry Central University. Indeed, I am very happy to see Bharati Mohan virtually. He is one of my lab mate. Both work together with Professor C.S. Sunandana in Central University of Hyderabad. Now, I request Shainita to introduce Dr. Bharati Mohan to the delegates. Over to Shainita.
Now I introduce our next speaker, Dr. D. Bharati Mohan Ram. He is an assistant professor in Department of Physics, School of Physical, Chemical and Applied Sciences, Pondicherry University from August 2010 onwards. He has done his PhD in Physics, School of Physics, University of Hyderabad between 2002 December to August 2007. He has received his postdoctoral fellowship from Department of Mechanical Engineering, University of Coimbra, Portugal between February 2007 to December 2009. He has also received his postdoctoral fellowship from Department of Mechanical Engineering, Korea, Advanced Institute of Science and Technology, South Korea, between Jan 2010 to August 2010. His area of specific activity includes nanoplasmonic biosensors and plasmonic solar cells. He has received many awards and recognitions like Young Scientist Award from DST Government of India in 2012, Brain Korea 21 South Korea Award in January 2010, European Fellowship by European Union between January 2007 to 2009, Research Fellowship under University with Potential for Excellence by University of Hyderabad between 2003 to 2006. His PhD thesis work published in the Handbook of Condensed Matter, Semiconductors, Spinjor. He has participated in many international meetings at Portugal, Belgium, Germany, France, Spain, Slovenia, Belarus, and South Korea. He has approximately one project received from UGC and DST. He has 54 international publications and he has participated in 75 conferences. He has also participated in 25 invited talks. He has got presently three ongoing PhD students and 2 PhD students have been awarded so far. He is a reviewer of ACS, IOP, Elsewhere, and Spinger. Now I request sir to deliver his talk.
हा दर्शू हा काढ की थांबा देते देते हा. माझा क्लास चाललेला तेव्हा.
शिवा गारू,
Yes, sir.
हो छोटीच काढ.
Bharti Mohan, sir, we can't hear you.
Bharati Mohan, your volume is very low. You can increase your volume, I think. Shiva garu, could you please help Bharati? Yeah, screen is okay. And Bharati, can you speak once? It's very low. It's very low. Voice is not clear, very low. Shiva garu, could you please suggest him something?
Yes, sir. Bharti Mohan, sir, could you please remove the headphones and speak to the speak near the laptop? Sir, or else you use another headphone, sir.
Shiva garu, what's happening? He has got some problem with mic.
I think so, sir. Because we're getting a very low voice from the mic.
Yeah. Very low voice you are getting actually. Bharati, can you unmute once again and you plug in or increase the sound something? We are listening, but not clear voice. Yeah. You have to increase the sound, I feel. I don't know exactly, but
हा पप्पा.
No, sir, we can't hear you. Sir, do you have another set of my headphones?
हा घे, ते नको तिकडचं घे, ते ब्लू कलरचं.
No, sir, we can't hear you. We can hear you, but not with a clear voice. No, sir. Venkateshwar sir.
Yeah, please. Shiva Garu, tell me.
Sir, Bharti sir said he'll be connecting through another system.
Okay. Okay. No problem. No problem. We will wait. Okay.
Okay, sir.
చేవితే గాడి వరా
శివ గారు ఎవరిది ఆ వాయిస్ వేరే వాళ్ళది ఎవరిదో వస్తుంది? హలో Shiva Garu, could you please Shanti Ganapa, can you meet Shanti Ganapa? Shiva Garu,
Yes, sir. Yes, sir. 1 second.
can
Give
you
me this.
meet Shanti Ganapa?
హలో
Bharti Mohan sir, we can see your video.
Shiva Garu, what's happening? Are you in touch with Dr. Bharati?
We're talking, so I'm not getting any response from the sir.
He switched on the camera, I think, actually.
Yeah, sir. Sir, do you have Bharti Mohan sir's phone number?
Yeah, I'm calling him. I'll do that.
Okay.
It's better. It is better now. It's better now. You can share the screen. You can share the screen. Shiva Garu,
Sir,
there is some lag in the voice, I think. Am I right?
yes, sir.
For
I
me,
think Bharti Mohan sir doesn't have a good connection of internet.
okay, okay. So let him switch off the camera and share the screen. That may be the best option, I feel. No?
Yes.
Dr. Bharati, could you please switch off your camera video and share the screen so that your internet might be very good actually. That is what my idea. Okay.
Okay. Sir, Bharti Mohan sir, so we can see your screen, but we're not able to clearly listen your voice. Sir, do you have another set of earphones or headphones? I can't able to hear you what you're saying also, sir.
Hello. Yeah.
Bharati, now your voice is clear.
Okay.
Dr. Bharati, now your voice is a little more clear.
Little more clear? Okay.
It's little more clear.
Okay. Then I will do this thing. Uh, I will try to start. Can you hear me? Hello. Can you hear? Hello. Yeah, I'm using the mobile. Okay. Okay, I will start. But let's take interesting time when I start my PPT, what we are going to do. Okay? Can you hear now?
So we can see your screen, sir.
Is it clear? Can you see my PPT?
So voice is not clear again.
Is it clear now?
No, sir.
Some technical issue here. But on the inauguration day, I was speaking with Korea. At the time, it was fine. So I thought that everything was properly. So now I'm finding difficulty.
Hello, Bharati.
Yeah,
Uh,
let me just check. Can you hear me
uh,
now? Can you hear me
no.
now?
Yes,
Yes,
perfect.
sir. Yes, sir.
Perfect.
Okay. Now I am sharing my PPT also. But I am using
Okay.
Now we are listening two voices. You switch off one system and use only one system, Bharati.
Okay, fine.
Now,
Now,
Yeah,
how about
fantastic.
now? Okay. Now,
One you switch off, one you mute, other one you use, so that we are getting two voices from your system.
is it okay now? Okay.
Shiva, is it okay?
Sir, yes, sir.
Perfect, Bharati. Thank you.
Okay.
Please carry on.
If it is okay, then I will start the presentation.
Perfect. It's okay.
Sorry for the technical issues. Because I was thinking, from my side, it's fine.
Shiva, two voices are coming. Am I right? Mr. Shiva?
Yes, sir.
Two voices are coming. Bharati, could you please? Yeah, one he muted now. Okay, perfect. Please.
Okay. Is it okay now?
Yes, sir.
Yes.
Okay. Now I will start my talk. Okay. Thank you for giving me this opportunity. I'm very sorry for this technical fault from my side. It is already delayed by 10 minutes. I will try to wind up, in 1 hour to 1 hour 15 minutes time. The title of my presentation is Engineering of 2D and 3D Nanoplasmonic Thin Films based on biosensor applications. Based biosensor applications. And the plan of my talk goes like this. First, I will introduce the biosensors, which includes the classification and characteristics, the recent diagnostics test that has been done using biosensor device. Secondly, I will discuss about the SERS, is nothing but surface enhanced Raman spectroscopy, and where I will compare the surface enhanced Raman spectroscopy scattering with Raman scattering, and followed by the recent application in the medical field. Thirdly, I will discuss about the fabrication of 2D and 3D CRS substrate that is fabricated in our lab, and the thin film deposition technique that is used. And I will also discuss about some of the physical properties we have used to study the 2D and 3D CRS substrate. Finally, I will discuss about the biomolecular reduction and the reusability by photocatalytic reaction. In this case, we have used two different biomolecules. One is a DNA, other one is a hemoglobin. All right. Can you see my next slide? Yes.
Yes, sir.
Okay. Perfect. I am using both PC and mobile phone. So I don't know how it works now, but it is working to some extent. So therefore, you could see some echo from my computer also. So if my voice is not having echo, it would be great for me. So now I will talk about the introduction to the biosensor. Biosensor is a device which is capable of producing quantitative information of analytes using a combination of sensor system and transducer and electronic system. Now, the very first biosensor was invented in the year of 1950 by American biochemist L.C. Clark, and this biosensor is used to gauge oxygen in the blood, and the electrode was used in the sensor is named as a Clark electrode or oxygen electrode. So, what is the working principle of the biosensor? Mainly, the transducer is coated with a sensory layer. It is nothing but biological recognizing element. Then it binds with the analytes that to be detected, And there is a biological change that occurs at the surface of the transducer due to the binding, which then converted into electric signal by the transducer. Then it is further amplified and converted into electronic signal with the help of microprocessor. And then the intensity of the signal output depends on the number of analytes that binds to the enzyme. So there are a lot of applications in a variety of areas like drug delivery, disease detection, environmental monitoring, soil quality testing, food quality monitoring, toxins of defense interest, water quality management, prosthetic devices, and drug delivery. So biosensors are classified into 4 types. One is the electrochemical sensors, and this is further classified into amperometric, potentiometric, imperimetric, and voltammetric. In physical biosensors, which is classified into piezoelectric, thermoelectric, and so on. Optical biosensors works on 3 different principles. One is by surface Passon resonance. It is called surface Passon resonance based biosensors. The other one is by means of scattering and emission called spectroscopy techniques. Wearable biosensors classified into several categories. I have mentioned here 3 of them. One is the smartwatches, second smart shirts, the third tattoos. Now the very advanced technique now recently available, and being tested. It is nothing but a contact lens. It is having a sensory layer which will have the capability to measure the amount of the glucose level from the tear fluid.
Bharti Mohan sir, sorry to interrupt. Can you make the PPT in presentation mode?
Okay. Yes.
Yes, sir. Continue.
Okay, fine. And now, one of the very important biosensors that we everyday use is nothing but a glucometer. It is also called a glucose sensor. This glucose sensor works exactly based on the principle that is invented by a scientist called Elsie Clark. Now, we need to look at the working principle of this biosensor. Each strip that we use in the glucometer has an enzyme layer. It is called glucose oxidase. And this enzyme reacts with the glucose that is present in the blood sample and creates gluconic acid. When this gluconic acid reacts with another chemical called ferricyanide, then it forms ferrocyanide. Once the ferrocyanide has been created, then this device runs an electronic current through the blood sample on the strip. So the current is then able to read the ferrocyanide and determine how much of glucose molecule that is present in the blood sample. So the number is then displayed on the screen of the glucose testing meter. So this is how the glucometer works. We need to, before we make a device, we need to set certain characteristics for this biosensor. There are 6 different characteristics which are very, very important before you make any device. The first one is the linearity. It is nothing but the concentration range over which the sensitivity of the sensor is good. The second is the response time. It is nothing but the time that is required to come to an equilibrium for having 95% of the response. And accuracy should be better without having any error. And the reproducibility is nothing but there should not be any change in the reading even after the multiple time of measurement. Sensitivity or LOD, nothing but limit of detection. We should have, if we make any device, we should achieve the lowest detectable value even down to the femtomolarity range. Coming to the specificity of the biosensor, the sensor layer that you deposit on transducer, which should be able to specifically attach the analytes in which we are interested. And finally, coming to the lifetime of the biosensor must be improved in a better manner. So these are the important characteristics. Now, coming to the detection of the disease. Suppose if a person is having a disease, our immune system produces a lot of antibodies, which will fight against the antigen to neutralize the antigens. So what are the best way to test or diagnose the disease of a human? One is the nucleic acid based test, the other one is the antigen or antibody test. The nucleic acid test is a technique used to detect a particular DNA or RNA sequence, helping to identify a particular species or subspecies of organisms, often called virus, a bacteria that acts as a pathogen in the blood, tissue, or urine. So coming to the detection of antigen or antibody. As I told you, our immune system produces a lot of antibodies which will attach to the antigen. The antigen which present at the surface of any bacteria or virus. Okay, so now each Y-shaped arm contains, that is from the antibody, contains paratopes that binds to the epitope of the antigen surface. So with the help of this binding, antigens are neutralized. So disease is cured or diagnosed. So I would like to discuss about the most important techniques that is available for diagnosing the disease. One is ELISA, other one is RT-PCR. RT-PCR is nothing but real-time polymerase chain reaction. ELISA is nothing but enzyme-linked immunosorbent assay. ELISA, these two are having an advantage as well as disadvantage. When it comes to the detection of molecule using ELISA, it detects only a protein with a specific antibody, whereas RT-PCR detects directly the DNA of a specific oligonucleotides. ELISA doesn't give you any quantitative information, whereas RT-PCR gives semi-quantitative information. If you look at how much time it takes for ELISA to detect disease, it takes about few weeks, whereas if you take RT-PCR, it takes few hours or in a maximum about a day to get the results. The handling requires, for example, in case of ELISA, it requires a limited training in a standard lab setting, whereas RT-PCR required moderate training, and it should be done in a specifically designed lab. The advantage of the ELISA, it is a cheap and quick way of testing pathogens in food. But whereas when it comes to RT-PCR, it is very accurate to test directly the DNA of the pathogens and highly sensitive, which can detect picogram to femtogram of analytes. And it requires only a small sample volume, and the test is reproducible, and it also provides an option for multiplexing. What are the disadvantages? If you look at for ELISA, suppose if you are heating the food, pathogens are killed, and ELISA gives only the information about the antigen. So once the pathogen is killed, then there is no information about the disease. But whereas if you take RT-PCR, since it is directly detecting the DNA, so there is no issue. So it is highly popular compared to the ELISA. However, RT-PCR is expensive compared to the ELISA. So maybe RT-PCR is the best at present, but due to the following difficulties such as it requires skilled workers with a high level of analytical expertise, and also it requires tedious sample pre-treatments, and which is not suitable for to perform on-site. So we all need a new technique in future. So that new technique should be a rapid testing, should have a rapid testing method. It should be ultra sensitive, and it should also give different fingerprinting which correspond to the different analytes. And it should also be a non-destructive analysis. And main importantly, this device is supposed to be used for on-site monitoring. So what is alternate technique that is available? Maybe CERS-based biosensor could be an alternate technique. Before I discuss about CERS-based biosensor, I will just discuss about, I'll just give introduction about the nanoplasmonics. What is nanoplasmonic? It is one of the very important component of nano-optics, which refers to optics at nanoscale. And the subject of the nanoplasmonics is nothing but the study of the optical properties of metal nanoparticles, which is determined by electron oscillations around a crystal lattice. There are two main important factors which are responsible for this nanoplasmonics. One is the localization and enhancement of optical fields, which can occur at nanoscale substance, and two, the frequencies of metal nanoparticle oscillations located in the optical spectrum. So the coexistence of these two factors causes an interesting physics, which forms a basis for numerous applications. So one of the very important technique that we use for the self-based biosensor is a Raman spectrometer. As all we know about the Raman effect, which is discovered by Sir C.V. Raman, who received the Nobel Prize in the year of 1930 for physics. What is Raman scattering? If a light is incident on a molecule, and most of the light is scattered by the molecule, that will have the wavelength which will be equal to the wavelength of the incident light, and that scattering is known as a Rayleigh scattering. But part of the light scattered, which will have a change in the wavelength, is called Raman scattering. There are two Raman scattering, one is the Stokes, the other one is the anti-Stokes. If the wavelength of the scattered light is greater than the wavelength of the incident light, then we call it as a Stokes scattering. If the wavelength of the scattered light is less than the wavelength of the laser light, then we call it as an anti-Stokes scattering. So the Raman spectra, they rely on only the Stokes scattering. So Raman spectroscopy will give the chemical structure of the molecule from the vibrational transition when the molecule undergoes a change in polarizability during the vibration. So the polarizability is nothing but ease of distorting electrons from its original position. So what is surface enhanced Raman spectroscopy? It is a sensitive technique, surface sensitive techniques, which amplify the Raman signal of the molecules that is absorbed on the metal nanoparticles. It has two mechanisms. One is a chemical enhancement, the other one is a electromagnetic enhancement. However, the mechanism for the enhancement of the Raman scattering of the probe molecule is in still debate. What is chemical enhancement? Chemical enhancement requires a chemical bond between the metal and analytes. So when metal is bond with analytes, then there is a charge transformation happening from metal to the molecule, and the charge transfer enhances the Raman scattering of the probe molecule. On the other hand, if you look at the electromagnetic enhancement, it do not require any chemical bond formation. And the Raman scattering of the probe molecule is enhanced through the excitation of localized surface plasmons. If you look at compare the enhancement factor, in both cases, enhancement factor for the electromagnetic enhancement is much higher. What is surface Boson resonance? Taking a single metal nanoparticle and it has electrons on the surface. When you incident with the light wave, some of the energy is absorbed by the metal nanoparticle. If the size of the nanoparticle is much less than the wavelength of the incident light, then what happens? The electrons get distorted away from its nuclei, which oscillates continuously with the incident wave, light wave, with the natural frequency called omega p. When this natural frequency matches with the incident light wave frequency, then it forms surface Boson resonance. The surface Boson resonance can be studied by using U-visible spectrophotometer. This figure shows the U-visible spectrophotometer of silver and gold nanoparticle. Silver nanoparticle exhibit a peak around 4 nanometer, whereas gold nanoparticle shows a peak around 525 nanometer. So the surface Boson resonance depends on the nature of the element, particle size and shape, and dielectric medium. So before we choose any metal for making a search-based biosensor, we must understand the plasmonic properties of the metal. So in order to understand the plasmonic properties of the metal, there are three important properties which are very important. One is the quality factor. It is a measure of how many oscillations and surface Boson undergoes before it decays, and also the associated increase in the local electric field amplitude surface when surface Bosons are resonantly existed by an external optical field. Secondly, the oscillator strength. It expresses the probability of an absorption of electromagnetic radiation in the transition between the energy levels of an atom. Thirdly, the model volume. It is nothing but Vm of surface Boson for any nanoparticle whose size must be less than the metal skin depth. It will be in the order of the volume of the particle itself, typically say 10 nanometer cube for any particle which is having a size about 10 nanometer. And model volume, which is very, very crucial for the sub-wavelength confinement of energy in nanoplasmonics. So what is unique about the silver? Silver is one of the very, very prominent material in case of in the field of nanoplasmonics because it supports SPR in the entire visible range to the near IR range. So SPR, but SPR depends on the dielectric function. The excitation cross section equation is derived from me theory. For any spherical particle, it is given as 24 pi square r cube epsilon m 3 pi 2 by lambda of epsilon I by epsilon r plus 2 epsilon m the whole square plus epsilon I square. If the resonance condition to be satisfied for any plasmonic material, the excitation cross section must be very high. It is very high only when the relative or the real part of the dielectric function of the material is negative. So it should, if you look at the denominator function of the bracketed function, the real part of the dielectric function should be the negative of the twice of the dielectric function of the medium. Only then the bracketed function approaches 0. When the bracketed function approaches 0, then the excitation cross section becomes high. So for any plasmonic material, the real part of the dielectric function should be negative, and same time the imaginary part should approach 0. If you look at the real part of the dielectric function of the three material, here I have shown, silver is having a negative value, whereas for silicon it is highly positive. At the same time, if you look at the imaginary part of the dielectric function of silver, which is approaching 0. Coming to the quality factor, which describes the strength of the surface Poisson resonance. And the equation is following: Q is equal to negative of the real part of the dielectric function by the imaginary part of the dielectric function. So the larger the quality factor, stronger the surface Poisson resonance. So if the quality factor must be greater than 10 for any good plasmonic material. And if you look at the graph here on the right hand side, quality factor is greater than 10 all through the region from UV to near IR region for the silver metal. Whereas if you look at for the aluminum, it is high only in the UV region. Therefore, aluminum can be used for the UV based sensor. And if you take gold and copper, this material can be used, and it will work best in the range from 600 nanometer to 1200 nanometer. So compared to all material, silver offers a very good property for the nanoplasmonics effect. So what is electromagnetic enhancement? If a molecule, say, is placed near the dimer, when the light is incident, it excites a surface Poisson resonance, which increases the localized electric field. And this localized electric field excites the Raman scattering of the probe molecules, which then again couples with the localized electric field and increases the Raman scattering to the level of e power 4. So this way, by localizing the electric field between the nanoparticles, and one can increase the Raman scattering of the probe molecules. What is the difference between the normal Raman scattering and surface enhanced Raman scattering? You take a glass substrate and you deposit a probe molecule, and you want to check the chemical structure of the probe molecule using Raman spectrometer. And you excite with the 488 nanometer, and it shows a Raman spectra with a very less intensity. Whereas if you take glass substrate and you deposit with silver nanoparticles, and then you drop cast the probe molecule, then you do the Raman scattering study, and it shows enhanced Raman scattering. In this way, just by depositing a probe molecule on the top of silver metal nanoparticles, one can enhance the Raman scattering of the probe molecules. So the enhanced matter can be calculated by using this equation. And there are some commercially available cell substrate made by the Ocean Optics USA. and the ocean optics, they have fabricated cell substrate, using a silver and gold metal nanostructure. And these substrates are used for the detection of narcotics, melamine, etc. So what are the different types of biomolecules that can be tested using this CERS-based biosensor? Albumin and globulin in a serum blood, DOX, which is nothing but a doxorubicin, which is a chemo drug called as an anthracycline, which will stop the growth of the cancer cell by blocking the topoisomerases, which is nothing but an enzyme. It also can detect influenza virus, swine flu, H1N1 1919. It also can detect E. coli, DNA, RNA, and hemoglobin. I would like to give some of the literature work that is available, which demonstrate the detection of the biomolecules. In this slide, I would like to show the detection of the albumin and globulin in serum blood. As we know, for any normal person, the A to G ratio must vary from 1.5 to 2.52:1. If there is any decrease in albumin without any decrease in globulins, and it causes severe liver disease, maldigestion, inflammation, and nephropathy, which is nothing but kidney disease. If there is an increase in the globulin without any increase in the albumin, then it causes inflammation, infection, multiple myeloma, it is nothing but a bone marrow cancer, and colorectal cancer. Here, people have taken a protein which contains some amount of albumin and globulin, and it is mixed with the silver nanoparticles, and Raman spectra is recorded on this material, and Raman spectra shows the level of albumin and globulin that is present in the serum blood. So just from the quantification of the albumin and globulin from the serum blood, one can easily understand the presence of the colorectal cancer in the proteins. The identification of neuroblastoma, which is again a type of cancer that forms in the nerve tissues, which is also detected by using a CERS substrate. So here, silver film, it acts as a CERS substrate, and then 3 samples have been collected, like tumor cells, which is directly from the tissue, and red blood cells and white blood cells, etc. So these 3 samples have been tested using Raman spectrometer, and you can see the tumor shell shows 2 predominant peaks at 730 and 1330 centimeter universe. So these 2 peaks correspond to a particular nucleic acids which is responsible for the growth of the tumor cells. So just by detecting the nucleic acid from the tumor cells, we can, by means of Raman spectrometer, one can understand the growth of the tumor cells in the tissue. So this CERS is again used for the real-time monitoring of drug loading and releasing. Initially, a gold nanoparticle is functionalized with the doxorubicin, and this functionalized gold nanoparticle has been injected inside the tissue, and the loading of the doxorubicin is continuously monitored with the help of the surface enhanced Raman spectroscopy. You can see as the time increases, the intensity of the Raman mode, which is observed at 460, which is decreased. As soon as it is released to the tissue, it completely disappears. After the release, the DOX chemo drug reacts with the cancer cell, and the interaction between the DOX and the cancer cell has been monitored with the help of fluorescence techniques. So with the combination of CERS and the fluorescence technique, one can continuously monitor the loading and interaction of the doxorubicin with cancer cells. So suppose if you are making a CERS-based biosensor, we have to design a compact size of sense-based biosensor. So you see the size of the Raman spectrophotometer, which is right now available, and it occupies a complete table space. But you want to miniaturize the sense-based biosensors, and this is a simple schematic picture like how one can develop a compact sense-based biosensor. So it requires 4 important components. One is a laser source, another one is a spectrometer, then optical fiber probe, and sample stage. So if you have all these 3, one can have a compact design. So it works based on the optical fiber based CERS device. Recently, scientists have developed a smartphone based CERS sensor, and in this device the CERS substrate have been fabricated on paper based substrate by drop casting the silver nanoparticles. 3 different concentration of rhodamine 6G have been prepared, from molarity ranging from 10⁻³ to -5, and this smartphone based CERS sensor is able to achieve the limited detection up to the level of 10⁻⁻5 molarity. So coming to the fabrication of 2D and 3D CERS substrates and the biomolecular detection. So to fabricate the CERS substrate, we have thermal evaporator and RF DC magnetron sputtering system and Teflon line autoclave used. Thermal evaporator used for the deposition of silver nanoparticles, whereas RF and DC magnetron sputtering system is used for depositing the bimetal, and Teflon line autoclave is used for growing the zinc oxide nanorods through hydrothermal reactions. So when you form a film using any deposition technique, there are 3 models involved. First one is a Walmer-Weber growth, or one can call it as an Highland growth model. In this model, the smallest stable clusters nucleate on the substrate and grow in 3 dimensions. Suppose if you want to deposit metal on SiO2, you form a island type of growth. The second model is a Frank van der Merwe model. It is also called as layer by layer growth. It is nothing but the smallest stable clusters condense tremendously in the 2 dimension, resulting in the formation of planar sheets. So if you are growing a silicon or titanium nitride on substrate like a silicon or MgO, then you form a layer instead of islands. The third model is a Stransky-Krasnov model, which is a combination of layer and islands. In this model, after forming 1 or more monolayers, the subsequent layer growth becomes unfavorable and forms island. For example, if you take indium, you want to deposit on silicon substrate, it forms layer as well as islands. If you are comparing the 2 different techniques, one is thermal evaporation, other one is sputtering. Sputtering is having a lot of advantages when it comes to the deposition of elements. The reason is that in sputtering, you can use a metal or dielectric. And other properties like uniformity, impurity, grain size, if you compare, the sputtering offers a better value. However, the sputtering technique is much expensive compared to thermal evaporator. Coming to the discussion of the fabrication and characterization of 2D CERS substrate, we have prepared the silver thin films using thermal evaporator with a thickness of around 10 nanometer. And we looked at the structural transition as a function of the temperature. This is the AFM image of the as prepared, as deposited silver film, which exhibit quasi-amorphous state. And once you anneal at 100 degrees Celsius, it converts into nanocrystalline state. And you can see the homogeneity in case of both size and shape, which is much better. And further, if you anneal the AFM, further if you anneal the film at 250 degrees Celsius, the nanocrystalline is converted into bulk status, where you can see formation of bigger size of nanoparticles and agglomeration is dominant. So the entire process takes place through the coalescence and Oswald ripening. We have calculated the particle size distribution as a function of annealing. You can see very clearly when you go from the quasi-amorphous state to the bulk, as you increase the annealing temperature, the number density of the particles increases. At the same time, the particle size becomes very narrow. On the other hand, if you look at in the quasi-amorphous state or in bulk state, the particle size is bigger and the number particle density is very small. Now, I am going to discuss about the fabrication of silver copper bimetal thin films. And the corresponding morphological and element analysis is shown here. The silver copper bimetal thin films have been prepared using confocal close sputtering technique. Here we used silver and copper as targets, and we have used the power supplies DC and RF. So DC is connected to the silver, whereas RF is connected to the copper target. The elemental composition of silver and copper has been varied by increasing the DC voltage of the silver, keeping the RF power constant for copper. And the atomic percentage of silver and copper has been investigated with the help of redox mapping, and we found that as you increase the DC voltage, the copper content decreases from 29% to 12%. The AFM image shows that the as deposited film as well as the annealed film, as you increase the annealing temperature, as you can see, the particle size and shape become more homogeneous, and when you decrease the copper content, what happens? The particle size become bigger and bigger, and the roughness also getting increased. The film roughness is calculated using AFM technique, just by scratching some portion of the film and scanning along this direction, and from the step size we can measure the thickness of the film. Now, we also studied some of the color change and SPR tuning in both silver and silver copper bimetal thin film. And we know the visible light varies from 380 to 750 nanometer. The physical change, the physical appearance of the films have been photographed for silver thin films, and you can see the as deposited film, silver film, starts with pink in color. As you increase the film thickness, it goes to dark blue. Then, when you anneal the same film at 400 degrees Celsius, it approaches green color. In case of silver copper bimetal thin film, you can see initially it starts with blue color and then it goes up to the bright yellow. It means you can see from this slide, just by changing the element, but from single to bimetal, it means you start with silver and you prepare bimetal, and changing the thickness and the annealing temperature, you can cover the region from 400 to 600 wavelength. So this way, one can tune the colors of the films by preparing the bimetals. And we also study the optical properties of the corresponding films. And you can see this is optical absorption of as deposited silver and silver copper bimetal thin films. The black one corresponds to the pink. And the SPR maxima is observed around 425. It is almost matching with the physical appearance of the color. And for the other films, you can see you have a long tail which is covering up to near ear region. If you anneal the same films at 400 degrees Celsius, you can get a very sharp SPR with a good homogeneity. Now there are three things noticed. One is long tail is disappeared. The second one is the SPR strength is increased, and you can see the intensity of the SPR is increased. And third one is the red shift. That means the blue one, if you look at, which correspond to the silver, which correspond to silver copper bimetal thin films annealed at 400 degree, and the SPR maxima is appearing around 500 nanometer. And we have analyzed these films and calculated particle size, surface roughness, and interparticle distance. Overall, the particle size is increasing with increasing film thickness and increasing the annealing temperature because of the increase in the crystallinity of the silver films. But if you look at in the case of silver copper bimetal thin films, it is very much confusing at a very lower thickness. In case of fast deposit and 200 degree annealed film, but when you anneal the film at 400 degrees Celsius, there is a systematic increase in the particle size, which goes from 30 nanometer to 85 nanometer. Coming to the surface roughness and interparticle distance, there is no much change in the surface roughness of pure silver film. But whereas when it comes to silver copper metal bimetal thin films, it systematically increases from 2.7 nanometer to 13.2 nanometer, and the films are annealed at 400 degrees Celsius. It comes to the interparticle distance. As Depart's film shows the interparticle distance of very small, like it varies from 4 to 7 nanometer. When you anneal the film at 400 degrees Celsius, there is a systematic increase, from 6 to 15 nanometer. It is the same in case of silver copper bimetal thin film also. One of the major problem with the silver is the oxidation. So we need to study the surface oxidation of the thin film. So we have used the Raman spectroscopy in order to understand the chemical structure of the silver thin film. And we found that there is a peak at 234, which corresponds to the stretching mode of silver oxide, confirming the formation of silver oxide. And the formation of silver oxide is become much greater when you increase the film thickness and when the films are annealed at higher temperature, like 250 degrees Celsius. So how to avoid this oxidation of silver film? So what we have done, we made a protective layer on top of silver and try to look at the oxidation of silver film. So initially we prepared a thin film of silver with 11 nanometer thickness, and the thickness is measured from the cross-sectional image using FESEM. And then the same film is annealed at 400 degrees Celsius, and you can see a homogeneity in the particle size and shape. After this, we have coated this film with Al2O3. The thickness of the Al2O3 will be ranging from 3 to 5 nanometer. Then we have done this optical absorption study for all these films. As you can see, the as deposit silver film shows a peak at 478, a very less intense SPR, maxima at 478. But when you anneal the same film at 400 degrees Celsius, there is a blue shift because of the nanocrystallinity. Then, after keeping for some time, and we waited for 4 weeks time, then we recorded the optical absorption, and you can see after 4 weeks, the SPR completely suppressed. Whereas if you look at the film that is coated with Al2O3, and it is stored for 4 week time, there is no much change in the SPR maxima as well as in the full width of maximum of the SPR. So this way, one can protect the silver with the Al2O3 layer to avoid the oxidation. Then how to prepare the 3D substrate? We have fabricated 3 different 3D hybrid structure, and here I am giving the schematic picture of the fabrication of silver-based 3D hybrid structure. Initially, we have taken glass substrate and we deposited silver thin film by using a thermal evaporator and annealed at 100 degrees Celsius. You get nanoparticles, and then this substrate has been used to grow the zinc oxide nanorod using hydrothermal process, and you can see that the zinc oxide nanorods grown vertically. And after this, we have deposited the zinc oxide nanorods with 3 different metals. One is silver, another one is platinum, another one is gold. Now you can see the silver coated substrate is called 2D substrate, whereas silver and platinum and gold decorated with decorated on zinc oxide nanorods called 3D substrates. Now let us understand the growth kinetics of the zinc oxide nanorods and with the effect of the seed layers annealing temperature. The growth of the zinc oxide nanorods is understood on a different platform. For example, the as deposited film is in a quasi-amorphous state. So when you grow zinc oxide nanorods through hydrothermal process on as deposited silver film, you grow vertical zinc oxide nanorods, but they are loosely densed. If you do the same, if you grow the zinc oxide nanorods on nanocrystalline platform, you get a very high dense and with the same aspect ratio of zinc oxide nanorods on the nanocrystalline substrate. If you grow the zinc oxide nanorods on bulk film, then you can see a loosely densed and angled growth of the zinc oxide nanorods. So again, we have calculated the particle size and interparticle distance of the silver nanoparticles and gold and platinum decorated on zinc oxide nanorods, and the particle size of silver ranges from 10 to 30 nanometer for silver, whereas in case of gold and platinum, it varies from 6 to 50 nanometer. Also, the interparticle gap has been calculated, and it ranges from 5 to 9 nanometer for the case of silver, and it is 2 to 5 nanometer for the case of silver and platinum. The reason for the bigger particle size and big higher interparticle gap for the case of silver, the silver is deposited using a thermal evaporator, whereas gold and platinum is deposited using sputtering techniques. So therefore, you could see some change in the particle size as well as in the interparticle gap. So this picture gives clear information about the bottom layer as well as the zinc oxide that is grown on the silver flame. So from TM image, we understood the formation of the hybrid structure of silver zinc oxide phase, and the crystals from crystal structure, that is from HRTM image, has been understood, and further it confirms the presence of the silver on the surface of zinc oxide phase. Another aspect is called hydrophobicity. The hydrophobicity is much better in case of 3D structure. You can see the schematic diagram. If you take a 2D substrate, on top if you drop cast analytes, it covers a larger area on the 2D substrate. But whereas if you take 3D substrate, if you drop cast analytes, it confined into a small area. So this is the advantages of the 3D substrate. So the confinement of pro molecules, it will help to increase the Raman scattering of the pro molecule. The hydrophobicity is measured using a water contact angle measurement study, and it is found that it is found to be 169 degree in case of silver based hybrid structure, which decreases to 152 for gold and platinum based hybrid structure. So without the decoration, if you measure the contact angle, it is found to be 144 degree. So the hybrid structure, 3D hybrid structure, shows the contact angle which is greater than 150 degrees. Therefore, we can call it as a super hydrophobic surface. And we know that lotus leaf having a hydrophobicity of 147 degree. So our material is having a better hydrophobicity compared to the lotus leaf. The enhancement of the Raman scattering can be explained through the Schottky barrier formation. When you are depositing a silver or gold or platinum on the surface of zinc oxide nanorods, there is a formation of Schottky barrier between the metal and semiconductor, because zinc oxide is a semiconducting material, silver is a metal. So when you make a contact, there is a Schottky barrier formation. So because of the Schottky barrier formation, there is a charge transfer from the metal to the semiconductor. So through the charge transfer, one can enhance the localized electric field at the contact of the zinc oxide and silver. And this enhancement of the localized electric field is higher in case of silver compared to the gold because of the different Fermi energy level. The homogeneity of the cell substrate has been tested using Raman mapping. The concentration of rhodanine 6G is 10 power minus 6 molarity. The peak in which we are interested is 1650 centimeter inverse. The homogeneity of the adsorption of the biomolecules, which is very, very important when it comes to the sensitivity of the cell substrate. So the homogeneity is studied using rhodanine 6G molecules on our cell substrate. And this picture shows a very good homogeneity of the cell substrate. We calculate the 3D enhancement factor and the 2D enhancement factor, from 2D and 3D substrates respectively, by preparing rhodanine 6G with a concentration of 10 power minus 5 molarity. And if you look at the enhancement factor in case of a 3D hybrid structure, it is 45-fold times. And that is observed in case of silver-based structure. Whereas in case of silver, gold, and platinum, it is about 18 and 4 respectively. And the limit of detection has been calculated for all three different hybrid structure. And in case of silver, we achieved LOD of about 10 power minus 18 molarity, whereas in case of gold and platinum, it is about 10 power minus 16 minus 12 molarity respectively. So the silver-based hybrid structure gives the maximum enhancement and better LOD. Then we use these substrates for the detection of hemoglobin, and the hemoglobin we have prepared with the drop volume of 10 microliter drop casted on cell substrate like silver-based 3D structure. Excited, we used Raman spectrometer with the excitation wavelength of 540 nanometer, and the power supplied 2.5 milliwatt and acquisition time of 6 seconds. And this hemoglobin is prepared with various concentration. As you can see, it is prepared from 10 power minus 4 molarity to 10 power minus 7 molarity to study the limit of detection. And as you can see that the Raman modes of the hemoglobin decreases with the decreasing the concentration. So the limit of detection of the hemoglobin on silver-based substrate is found to be 10 power minus 7 molarity. And then we also tested with lambda DNA with a drop volume of 10 microliter. And in this case, we used gold-based hybrid nanostructure. The wavelength of the laser light was 633 nanometer, and the power that we applied 0.085 watt, and acquisition time is 90 seconds. We prepared DNA with a different concentration ranging from 500 nanogram per microliter to the 10 nanogram microliter. And you can see that Raman spectra is obtained up to the concentration of 10 nanogram per microliter, which means the limit of detection is up to 10 nanogram per microliter for the case of gold zinc oxide hybrid nanostructure. So we have done the reusability test by applying the photocatalytic reaction. And in this case, what we have done, after drop casting the pro molecules and recording the cell spectra, we dipped this entire thing in the water and shined with the light with the wavelength of 257 nanometer to wash away the entire rhodanine 6G molecules from the substrate. and the substrate become again fresh. And this, with the help of the photocatalytic reaction, we can reuse the same substrate multiple times. So this is the advantages of the 3D hybrid nanostructure. What is the mechanism of the photocatalytic reaction here? All we know that zinc oxide is a very good material for the photocatalytic reaction. And once you excite with the light, electron goes from valence band to the conduction band. Electrons combines with the oxygen, or it gets trapped with oxygen and forms superoxide, whereas holes in the valence band reacts with water, forms hydroxyl ions. So the formation of hydroxyl and superoxide radicals will degrade the organic molecules into the inorganic molecules. So this way, the rhodamer 6G that is drop casted on the CERS platform has been degraded with the help of photocatalytic degradation technique. And you can see here, for the different times of degradation, we have recorded the Raman spectra on both silver-based and gold-based nanostructure. As you increase the time, and you can see at 40 minutes time, after 40 minutes time, the Raman modes of the rhodamer 6G is completely disappeared. And it is again happening in same in case of gold-based nanostructure. So the photocatalytic degradation is described by a Langmuir-Hinshelwood mechanism, which follows a pseudo first order kinetics. The degradation rate constant is calculated from these two plots, straight line, and it is found to be 0.1928 per minute for silver, and for gold it is about 0.1303. So the degradation rate constant is much better in case of silver as compared to the gold-based hybrid structure. So we have done the reusability study, and we have tested this same substrate, and we have detected the rhodamer 6G up to the cycles of 8 times. So you can see this is the first detection, and after photocatalytic reaction, the rhodamer 6G is completely removed, and then again the rhodamer 6G was drop casted, and then again we detected with the same intensity, and this has been repeated continuously for 8 cycle, and we found that even after the 8th cycle, you can reuse the same substrate to detect the rhodamer 6G probe molecule. And this has been verified again with the gold-based hybrid nanostructure. The degradation study is again done on these biomolecules. So we tested with hemoglobin as well as with the lambda DNA, and you can see very clearly that when the degradation time increases, the Raman modes of the hemoglobin and the lambda DNA get suppressed, and it completely disappears after 100 minutes of degradation time. And the degradation constant is again calculated for these two, and it is found that silver is offering a better degradation constant compared to the gold-based nano substrate. And again, the reusability is done in case of hemoglobin and the lambda DNA. We have tested up to the cycle of 4 times, and we found that even after 4 times of testing, we are able to reuse the same substrate. So now conclusions. Scientists started to apply this Raman spectrometer for the COVID-19 testing. CERS will be an alternative method in the field of physics which is related to the interaction of the light and matter. This test will be a much more precise and reliable method to detect infection. This is a physics-based testing approach, would be ultra-fast, inexpensive, ultra-sensitive, and highly specific, and deliver lower percentage of the false negatives. Also, a portable Raman equipment could allow the test to be used widely in rural and remote communities or in point-of-care sessions in schools, factories, and community centers, in addition to the traditional testing sites. Finally, I would like to acknowledge my students, Dr. Anil Kumar Pal, whose contribution is much in this presentation. All the animation is all the animation work is done by Dr. Anil Kumar Pal, now who is a postdoc fellow in UK, and part of the work from Mr. Nadagopal has been included in the presentation. So I have to thank particularly these two students, to help in my presentation. Also, I would like to thank my collaborators, Professor S. Umapati and Dr. Gautam Chandra, who is from IIC Bangalore. Regarding the Raman spectroscopy measurements, and I thank Center for Nanoscience Engineering, IIC Bangalore, towards some of the characterization techniques, and Dr. R. Prashant and Mr. Jyotish from Center for Green Energy and Technology from Carnegie University. They helped towards measurement of the contact angle study. And Mr. Sudhakar Pagal and Dr. Prashant Kenjapa from Department of Biotechnology, Pondicherry University, now for the preparation of the biomolecules and studying the biomolecules using this substrate. Finally, I thank the funding agencies like UGC, DST, and startup grant from Pondicherry University. Thank you for patiently listening, and I'm very sorry for this, technical fault from my side. I do not know how much I have delivered because I have connected my microphone to my mobile and speaking through mobile, but presenting the PPT from my PC. I don't know how it works. I hope you could listen my talk, and thank you very much for giving me this opportunity.
Fanny ma'am, are you there?
Yes, sir, I'm there.
Ma'am, do you want to ask sir any questions? You can ask.
So sorry to say this, dear participants, I actually lost the internet connection for 10 minutes, so I'm unable to read any questions in the chat box. If you want to ask any questions, please put them in the chat box right now. To all the participants, I request once again to put your questions in the chat box so that I'll pass the questions. Bharti Mohan sir, are you there, sir?
Yes, yes.
So today's first question is, if we increase the film thickness, how the band gap of the material will vary? I mean, whether it will increase or decrease. Please explain the mechanism involved in it.
Band gap. You're talking about the band gap, right?
Yes, sir. Do you want me to repeat the question?
No problem. See, metal do not have any band gap. We need to look at only the surface Plasmon resonance of the metal nanoparticles. The surface Plasmon resonance of the metal nanoparticles, if you look, you want to go with the optical spectrophotometer. So optical spectrophotometer gives information about the surface Western resonance of any metal thin films. And as you increase the thickness of the metal thin films, take silver nanoparticles or silver thin films, the SPR shift, there is a SPR shift. Okay, it may be a blue shift or red shift, depending on with what state you have started with. For example, in our case, the as deposited silver film exhibit quasi-amorphous state. So when you anneal the film or when you increase the thickness, what happens? The quasi-amorphous state is converted into the bulk state. So in the transition, the film can go to the nanocrystalline nature also. Suppose if the film is converted into nanocrystalline, there will be a blue shift because of the decrease in the particle size and the shape. But if you go from the nanocrystalline to bulk, the particle size again increases. So when the particle size decreases, there is a blue shift in the SPR, whereas when the particle size increases, there is a red shift in the SPR. So one need to only worry about the SPR maximum of the metal thin films when you prepare. So as you increase the film thickness, normally the particle size increases. Therefore, there is a red shift in the SPR. Yeah, this is my answer.
Sir, will the same be the answer even for the semiconductors, or the behavior will change in case of semiconductors?
Sorry, can you repeat? I'm not able to listen.
Will the same phenomena be for semiconductors, or will there be some other phenomena for semiconductors?
Yes.
Sir?
Yes. See, if you take a semiconducting material, and if you prepare a quantum dots of semiconducting material, same thing happens. But here, it is a band gap. So band gap is measured, is a gap between the valence band and the conduction band, right? The energy band gap is nothing but the energy difference between the valence band and the conduction band. So here, as you increase the size of the quantum dots of semiconducting material, again, there is a blue shift, sorry, increase the size of the quantum dots, there is a red shift. Okay, because if you increase the quantum dots, there is a shrinking of the band gap. So because of the shrinking of band gap, there is a there is some connection problem. Sorry, I am repeating again. Can you hear me?
Sir, we can clearly hear you.
I'll repeat again. Okay, so when the size of the quantum dots increases, there is a red shift, which means that the energy is very high. So energy is high means the band gap is, if the size of the quantum dots decreases, there is an expansion in the energy band gap. So there is an increase in the band gap. So the increase in the band gap is nothing but the blue shift, and decrease in band gap is nothing but the red shift. And the same thing happens in case of semiconductor. But in case of semiconductor, it is a band gap. But in case of metal nanoparticle, it is a surface Western resonance. Okay, so you need to understand the difference between the surface Western resonance with the energy band gap. Thank you. Any more questions?
Thank you, sir, for answering the questions. We are done with the questions. It was your session by you.
Thank you.
Thank you all the participants for joining this session. We are so sorry that there was some technical issues in between. Now I'll be sharing the feedback link in this group as well, Telegram and WhatsApp. Please give us feedback. Thank you, and let's join the event tomorrow once again. Bye.
I am ending the event.
Bhati Mohan sir, are you there?
Yes.
Actually, I got a message that there are some questions from NanoTV. So can you hold on for some 2 minutes, sir? I'll just check them out.
Yeah. Can you please tell?
Yes, sir. Shiva, sir, can you see some questions from Nano TV? Actually, I'm unable to find them.
Can't see any questions.
I'm not finding any questions over here. Are you able to see some questions in the YouTube chat box?
Yes, sir.
So
Okay.
I have to see some questions here. Raman spectroscopy technique will help. How this Raman spectroscopy will help in COVID-19 direction? That is one question. Another question, which type of capping agent can be used for reducing the size of the particle? Yeah. So I can read some question here. Can I answer for those questions?
Yes, sir, you can. Shall you want me to question question?
I can go to the chat box and answer.
Okay, sir, you can please go ahead.
Yes. And then later I have answered the questions, and then how the thickness affects on the sensitivity of the sensor. So this is one of the questions. So basically, thickness is one of the very important property when it comes to making a sensor. But what we should know is that what type of pro molecules in which we are interested. As we know that the thickness is one of the property to tune the surface Preston resonance of the metal nanoparticle. So if you are able to tune the SPR peak, and if there is a frequency matching, between the SPR peak and the Raman scattering of pro molecules, then you can enhance the sensitivity of the source-based biosensors. So thickness is the one of the property which can help you to tune the SPR. So with the help of the frequency matching between the SPR and the Raman scattering of pro molecules, we can further enhance the sensitivity of the sensor device. And the next question is, what is the effect on band gap when we go from bulk to nano? So as I told you, when you go from the bulk to nano, the band gap, there is a blue shift. Okay, so there is a blue shift. So blue shift in the sense it is going to the higher energy region. Higher energy means the band gap is larger. So as you go from the bulk to nano, the band gap increases. Therefore, there is a blue shift observed in the semiconducting nanoparticle. The same thing is true in case of SPR also. When the size of the nanoparticle increases, the SPR is blue shifted. When you decrease the size of the nanoparticle, the SPR is blue shifted. When you increase the size of the nanoparticles, the SPR is red shifted. How we reduce the size of the particle during the synthesis process? So it is again by like what we have done. We have fabricated silver and silver sub materials with the help of thermal evaporator and other technique called sputtering. These two techniques are called thin film techniques. So we have used two different parameters. One is the thickness, the other one is annealing temperature. So just by varying the thickness and annealing temperature, we were able to tune or control the particle size of the metal nanoparticle. So as I told you, when you start with quasi-amorphous state, which is as deposited, which is as deposited thin films, and when you anneal this film at 100 degrees Celsius, and it becomes nanocrystalline. But if you anneal again at very high temperature, again it goes to the bulk. So the tuning, tuning in terms of reducing, in terms of decreasing the particle size, or in obtaining the smallest particle size, it depends on the thickness of the films that is coated. If you increase the film thickness, for example, from 5 nanometer to 50 nanometer, and if you anneal such films at a very high temperature, and it may not go to the nanocrystalline state, it may immediately go to the bulk state. So therefore, in order to obtain a very smallest particle, it depends on the film thickness as well as the annealing temperature. Next question. How we reduce the size of the particles during the synthesis? Okay. Which types of capping agents can be used for reducing the size of the particle? Okay, we have not used any capping agent to reduce the particle size here. So we have only varied the thickness and annealing temperature to reduce the particle size. Suppose if you are preparing a silver metal nanoparticles using PVP or CTAP, I would suggest you to go with the CTAP. CTAP, you can go for the preparation of the nanoparticles, silver nanoparticles using PVP as well. But you can try with both. But our experience tells you that in both cases, you can reduce the particle size. Both will act as a capping agent for controlling the particle size. And CTAP can be a better capping agent for controlling the particle size compared to the PVP. So if you prepare any materials with CTAP, it can control the particle size. So lecture was nice, and in COVID-19 direction, how this Raman spectroscopy technique will help. See, when it comes to the Raman spectroscopy technique, what I have discussed about is the label-free detection. In the label-free detection, we don't use any antigen to attach with the analyte. Whatever the analytes I was talking about, hemoglobin and DNA, and as a standard pro molecule, which is rhodamin 16. So this attachment is a physical attachment in our cell substrate. So if you are able to increase the sensitivity, if you want to increase the sensitivity of the detection, you need to have a very good platform. And in our experience, what we found that forming a 3D structure enhances the Raman scattering of the pro molecules. So people started, working on this, but people have not done much on this COVID-19 detection. But in future, if you are developing a sensor device based on CIRS-based one, then it will be a better or alternating technique. It will be an alternating technique for the ELISA or RT-PCR. So still the research has been started and people have started working on this. And so far there is no outcome as far as the COVID-19 detection from this Raman spectroscopy technique. But it is only happening at the research scale. Yeah. Okay, that's all. I have answered for all the questions which has been put up here.
Sir, for sparing your valuable time once again, holding back yourself and answering all the questions that were before. I once again thank you on behalf of KVR, sir, Center for Nanoscience and Technology, JNDU. Thank you so much.
This opportunity.
Okay,
Shall I
dear.
close the session?
Yeah, I have shared the feedback link, and once again, the feedback link will be posted on all the applications, I mean, all the Telegram WhatsApp groups. So, see you all tomorrow by 10 a.m. or 10 minutes, that is 9:50 a.m. Good day.